Okay, so good morning, everyone, and welcome back. We're into the second lecture of the course in the bioethical issues at the beginning of human life. And so always we begin with a little prayer in the name of the Father, the Son, and the Holy Spirit. Come, Holy Spirit, fill the hearts of your faithful and enkindle in us the fire of your divine love. Through Christ our Lord. Amen. All right, so at the beginning of this course, we're settling more the biological facts. So since you all have a varied background, for some people, this would be easier and kind of a review of studies. For other people, this may be a little more challenging, but it'll balance out when we get into the ethical analysis or the bioethical analysis. We need to establish the background. Before I get into that, one of you asked about the internships. I wanted to talk about the internship a little bit and leave it on the recording for others who are not here today. They can watch it or hear it. So the internship is one of the 8 courses, and it is 4 credits, and it is 150 hours of volunteer work. If you get paid for it, then more power to you, you know, but few people or few institutions pay for internships anyway, but if it's paid, it's fine. Doesn't matter. The important thing is to get it done. So this is how it works. It's 150 hours total. And you'll actually register for that course in the fall of this year, but you don't have to wait until the fall. In fact, don't wait until the fall, because 150 hours, you can spread them throughout the whole year, all right? The calendar year, essentially from now until, I'll give you until the end of November, because then in December I have to put in grades. So it's a pass-fail course in the sense that you either meet 150 hours or you don't, all right? And this is how we work it. You can do an internship in whatever you like, as long as it's associated with bioethics. In other words, the idea is to start talking to people about bioethical issues. It could be either bioethical issues at the beginning of life or end of human life or in healthcare, or it could be environmental bioethics. And so basically, as you pick up knowledge throughout the courses, you start, that's kind of your toolkit that you will use for entering into dialogue with people, all right? And so depending on the setting, for example, you can do easy settings like the Respect Life Ministry, which they have about 5 different counseling centers between Broward County and Dade County to counsel pregnant moms to keep their babies and to help them along the way, encourage them and so forth. That's one. Or straight up in a hospital, do volunteer work there also. Or it could be, if you want to do environmental things, there's one place called Pelican Harbor, which I think it's in Coconut Grove area. They rescue injured wildlife. And it's in the Julia Tuttle, yeah, 41st Street. Oh, okay, so it's further north. I see, okay. All right, yes, one of my students 2 cohorts ago did her internship there, and one time they had a pelican who had a hook in its beak, and so they took out the thing, and then the pelican was in quarantine for a while for healing and so forth. And once the vet thought that the pelican was healed enough, then they released the pelican. And so she was in charge of the release. So you can do a number of things. It's up to you, really. It could be in, you use your imagination. It could be in a clinic. You can be with a medical doctor or something like that. The idea is to engage others in talking about bioethical issues. Another one that is wide open is at a parish level. You can talk to your pastor, for example, if you want to give talks. You can go to the CCD program, for example, with the children and talk to them about some of the bioethical issues, or in the adult catechesis, the parish, or you can just do a straight up talk, a conference on bioethical issues. Again, it can be an overview, or it can be more specific, like beginning of life, end of life. All right? So it's really quite varied, number one. Number two, you don't have to do the 150 hours in one particular setting or venue. You can switch around, all right? You can do 2 or 3, as many as you want, really. When you reach 150, that's the end of it for the course itself, all right? If you want to continue doing volunteer work, that's fine. You can go to high schools, for example, and give talks if you want, things like that, all right? But it's up to you. So I leave it to your creativity. If you're having difficulty finding a place to do an internship, I'll be happy to help you there. But try to find them on your own if you can. And then what I'll do is I'll send you a spreadsheet. And the spreadsheet, I can show you here for a moment. Let's see, let's go to the course. It's very simple. It has 2 documents, a spreadsheet where you keep a track of your time, and there's also a little form that one of the supervisors will just sign. Let's see. Turn off the lights here. Okay, so that's the spreadsheet. You can see it's very straightforward, very simple. I will send you this by email. And when you get it, just download it and save it and put your name on it, your first name or your last name on the title, all right? And here you put the date. Let's say this afternoon you're gonna do 3 hours at Pelican Harbor. So you put today's date and you put 3 here, 4 hours, all right? Round up to the nearest half hour in decimals. So if you do 3 hours and 25 minutes, that's 3.5 hours, all right? Right up to the nearest half hour. That's accurate enough. And then at the end of the internship, you just sum this column. And again, when you hit 150, you're done for the requirement. So each time you do some time, some hours, you just the date and here. I leave it up to you. If you want to keep this for your own records, you can then expand this column here and put comments or whatever. You can write whatever you want there. I'm not requiring it, but you can do it for your own records if you want, okay? Number one. Number two, again, the 150 don't have to be in the same place. You can switch around different places. But I do need one supervisor, maybe the place where you did the most hours, but not necessarily, just any one of them, to fill out this form, which is pretty straightforward also. It's a little scale of 1 to 10. Just 4 questions. Is the person being punctual? In other words, are you being professional about the internship? You know, are you being punctual, and are you following the instructions that they give you at the place and treating everybody else with respect and so forth? And then there's the final question here. Anything that the supervisor wants to tell me, typically they put very good comments here because they're all very happy that you're helping them, all right? And they send me this back. It's confidential because of the comments, and that also allows the supervisors to be open and frank about the whole thing. But this is just a paper trail, essentially, that if someone asked me to justify the internship or what kind of internship you did, I have something, I have a paper trail to back it up that you, in fact, did some internship someplace. And
sent this to us already?
I don't know, but I'll send it now. Okay. So here goes the name of the student, obviously not the signature of the student because you will not be given this form, but I'll send it to you so that you can then print it and take it to wherever you're going. The name of the mentor and the mentors.
You did something like on Earth Day, volunteer work.
Yes.
Would that be considered any?
Yes.
So more like active doing something rather than
Right.
discussion.
Right. No, you can be doing something, but as you do something, hopefully you won't be isolated. You'll be talking to other people about it. All right. And you can go to conferences. If you see some conferences, there is the the bioethics conference and groups that they do. There's a group that goes down to the Rockland Pine area in South Dade to pull out weeds and stuff like that. You know, that's.
Right. I was going to mention that sometimes I do volunteer work in some of these county reserves.
Yes.
And of course, usually I have to receive people and
Right.
training on the
Exactly.
ecosystem and the value of the environmental work they're doing.
That's it right there.
Watch out to them.
Exactly. So you're
Combination of
supervising a
physical work
group.
and supervision.
Exactly.
So that will count.
Absolutely.
Being
That will
AMSA,
count.
for example, being AMSA, I have a bunch of students that they come pick up trash and I
Right.
have to look after them. But
Right.
I-
And trash pickup day at the beach or one of the parks.
I love the tree.
Yeah. All that counts, and you can maybe introduce the issue of invasive species and so forth, which is a natural. And see, we're looking at it from the bioethical perspective that we need to do this, right? Because we have stewardship over creation. So really, it's very open, it's very creative, if you will. And hopefully, along the way, you'll be able to engage some people to think about the issues. Now, many times they're friendly people, sometimes they're challenging. For example, if you run into someone who is pro-physician-assisted suicide or pro-euthanasia, you know, to try to convince them why that's not a good idea. But at least to have the dialogue. The same with people who consider themselves just in favor of abortion or women's choice for abortion. Try to engage them and at least have a dialogue about it. Okay? So that's the internship. Okay? I do recommend that you start it as early as possible because those 150 hours will pick up soon, and if you leave it for the second half of the year, then you know, everybody gets busy.
You still receive masters if you don't pass the internship?
Well, it's one of the key courses, and so to the bottom line, no, it's not possible. Why? Because it's 4 credits, and I've designed the program so that it's 30 credits, which is the minimum for a master's. If you take out the 4, then you don't have the required number of credits for the master. I've kept it at 30 so that it's the least expensive. I could put more courses and more material, but then that makes the program more expensive. You see, so that's the thing there.
Would you, okay, I'm just wondering if there's also any viable chance of doing like an independent study on issues in lieu of or in addition to the internship? Because
Oh, you can do.
frankly, Father, I mean,
Yep.
with all due respect,
Yep.
I mean, I told you this when I signed up in the class, I am pro-choice, and I, you know, so I have no problem with other people respecting, and that's why I'm pro-choice.
Yeah.
So, like if, you know, or even physician-assisted suicide, naturally assisted death, whatever, I am strongly in favor of this, so.
Okay, we'll get there.
No, I understand, but what I'm saying is, I'm not sure, I mean, I can find volunteer talking about things, learning, that's not an
Right.
issue. But,
Mhm.
you know, I can see frankly making the argument that if I participate in pro-choice activities, that would be part of the internship.
Well,
I can see where you would not agree.
not necessarily. I
Okay.
mean, if that's your point of view, I am very eager to get into the nitty-gritty of it so that we can have a dialogue about it and see what happens.
Right, no,
Maybe we all, after we finish this course, we all become pro-choice, and then you convince us all that that's the way to go. You know,
But there's other issues, those familiar issues.
Exactly.
I understand that.
of
But
course.
that's what I'm saying is some of the issues, but again, I mean, honestly, last Sunday's New York Times, I just got that time this week, was an incredible article about gene switching,
Gene editing.
it's not really the editing, but it's
Okay.
basically taking parts of the DNA code in mosquitoes
Uh,
to change it so that they can't spread the malaria, then
Yeah.
I'll list the mosquito. But it was a fascinating
Basically,
article about-
transgenics. That's been
and it's
done.
using some CRISPR
quite a bit.
therapy.
Right.
And it was absolutely fascinating bioethical considerations. So,
Right.
I mean,
You can get into that. That's fine.
so like if,
Or you can get into the non-human bioethics.
well, but it really was because,
Uh-huh.
you know, could you do this something if somebody had the power to wipe out agricultural crops or beneficial insects?
Right.
Insects.
Exactly.
there's all sorts of
GMOs, some are
Right.
issues with GMOs. So that that generally is under the environmental course, which we
Right,
won't
well,
have
I just
until the
said, like,
summer, but you can get into it already.
If some of those
Mhm.
additional readings or sharing or making
Yeah.
people aware, if you might be a little more
Yeah, as long as,
flexible on the internship part.
well, no. I mean, the internship is very flexible. The point is that it should not just be studying and learning on your own, okay? Because that's more of the same. You want to be able to interact with others on that, to engage others in that dialogue, okay? To engage others in the dialogue, that's the whole idea. And even if it's just one person, it could even be informally with family or friends, for example.
Okay. Because we've been discussing some issues
Yeah.
and, you know,
That's fine.
some of my
As
Christian
long as-
colleagues.
Yeah, that's what I'm saying. The whole issue is to get other people to think about bioethical issues. Okay?
Okay,
Maggie.
so if I'm doing this in a Catholic setting, like the church, RCIA,
Right.
whatever.
Yep.
And I don't know the church's position on what, like the one you were just talking about sounds fascinating with the mosquitoes and stuff. So say we're talking about that.
Mhm.
I don't know enough to present the church's side of it, because I don't even know if the church has a side on it. They don't always have a side on it.
Exactly.
yet.
So for that one, you may want to wait until the summer until we have the course in May.
Okay.
It'll be May and half of June. Okay.
Okay, so when leading the discussion or you know,
Mhm.
facilitating it, whatever you
Yeah.
call it, I need to know the church's position on whatever it is, because
Yes.
that's kind of.
No, I don't think you do. I think
But you
need
can-
to say that there's, I mean, unless you say I'm teaching the church's position, but even if you just say, these are areas of concern that as Catholics,
Of course.
as citizens, as people, we should be.
Right, but we should have substance
Teaching,
in our debates and our discussions. So if we have our personal positions, that's okay at a personal level. But to apply it to this degree, I think that we need to have substance in our debates and not just being activists of a particular subject. I hope that it's something concrete, because I can go outside and start talking about whatever I want. But
Of
I think
course.
that
Well, that's the whole point.
you want some substance, so this is just hours that we'll put out just engaging.
No, obviously
Find
it
people's
should be
opinions and
it should be
my-
substantial. So
What is the order?
all right, so the beauty about graduate work is that we're dealing with responsible adults that will do something that is in conscience. But we also respect academic freedom and freedom of expression and so forth. Therefore, it's all in the argument, the power of the argument, right? The power of reasoning. Now, with regard, I can tell you, Maggie, that the church does not have very specific positions on environmental issues, all right? Even though we can take Laudato Si, for example. I don't know if you read Laudato Si yet, the Pope's encyclical on the environment from 2015. Yeah, you can download it for free anyway from the Vatican in whatever language you want, and we'll cover Laudato Si, various aspects of it, again, in the environmental course. But we certainly make a big distinction, the church makes a big distinction between human life issues and non-human life. So whereas we can do experiments on animals, for example, without their consent, right? We certainly cannot do experiments on humans without their consent and so forth. So, and even certain experiments that we would do on animals, we would never do on humans, right? So, I mean, there's already an intuition there just from our own Catholic background of where the church would stand on some issues. But the church has made many more declarations with regards to human life than with regards to non-human life.
And I mean, the same with like GMO foods and gene
Yes.
editing and all of that stuff.
Exactly.
I'm not sure the church has a position.
It doesn't. I can tell you it doesn't have a
I'm
position.
leading a discussion in the Catholic community. I
Yeah.
feel that I should know the church's position so that not
Right.
everybody's opinion is welcome.
Okay.
It's a discussion, but I have the position, the
Yeah.
supposition that the people who are going to come to a discussion are going to expect to
Yeah,
like
to get the
learn
Catholic
something.
version,
I'm not
yeah.
here, like to teach, just to sort of guide instruction.
Right, you can facilitate the discussion. So my suggestion, if you want to get into the environmental issues, start reading Laudato Si, all right? Or you could just put Pope Francis on the internet, Pope Francis,
Yeah.
right? Environment encyclical, those keywords should be sufficient, and you go to the official site of the Vatican and you can download it for for free, or you can buy it. It's about 150 pages in booklet form in whatever language. So we'll be using that one again in the summer course, but you can start reading up on it. In the meantime, you can get into other areas until you become more familiar or you feel more confident, let's say, speaking about environmental issues from the Catholic perspective.
Okay. One more thing.
Yeah.
The 150 hours.
Mm-hmm.
We had talked before, and it was very informal, so I don't want to hold you to anything, but
Right.
I want to be sure. All 150 hours don't have to be signed off by the mentor?
That's correct, because especially if you have in different venues, different places, the mentors are different, right?
Okay.
So I just need one mentor to sign off on the form, just
On
one.
all 150, so they're signing off on.
No, on that particular internship. That's what I'm saying. Ideally, it would be where you would do the most hours, but not necessarily. So hypothetically, if you're going to do stuff like that, you know, you're a catechist, right? You teach. Okay, so typically the pastor, or if you have a lay person or a deacon that is supervising all catechists in the parish or something like that, that's sufficient. Okay? Yeah. It's just so that I have some paper trail that at least one of the internships was supervised. But to do 100, that's what I'm saying, 150 hours in just, some people do it, you know, 150 hours in one particular place. But I want you to use it as broadly as possible.
I mean, if I go to lunch with my daughter and we talk about oral contraception for an hour and a half, that
Well,
counts, right?
If you get her to engage in it,
Oh, yes, of course.
yeah, then, yeah,
Let's
sure. Yeah.
So, yeah. Okay.
That's what I'm saying, you know, we're all responsible adults, and I trust that you're going to put the 2 hours there and not just when you're having lunch, but not talking about bioethical issues. Okay.
The period will be from the beginning of this year to the end?
Yes, from now, as I'm telling you, from now all the way to the end of November. So I'll give myself just one little week there of margin, but you have until the end of November. Basically, look at Thanksgiving as the limit, right? And if you want to include Thanksgiving Day and talk about controversial issues on your Thanksgiving dinner, be my guest. Okay, so as we speak then, might as well send you these so that you have them. And I can just go here and attach these two. All right. Internship. Cohort. There he goes. All right. Any other questions, comments about the internship?
For this class, Father, is there any specific book or material?
Oh, thanks for asking. Yes, the Ethical and Religious Directives for Catholic Healthcare Services, ERDs for short. It's in the syllabus, but let me pull that up. And you can just put into Google Ethical and Religious Directives. Okay, is that large enough? Ethical and Religious Directives. The full title is Ethical and Religious Directives for Catholic Healthcare Services. The title is almost longer than the booklet. All right. It's an official document from the U.S. Conference of Catholic Bishops. The latest edition is 2018. So you can go, for example, don't go to the first site here, that is the PDF, because you can see it's in 2009. That's the 5th edition. Now there's a new one. So go to this one that has the official website you see here, USCCB. Is that large enough? Can you see it? Oops. Let's see. United States Conference of Catholic Bishops. That's the official website of the bishops of the United States. So you go there. Okay, can you see that? Yeah, all right. So you see, it's a 6th edition. It was published last summer. And you have it in English or in Spanish. And you can download it for free. And there it is. Okay, it's got about 30 pages, but it's a booklet form. I happen to have one here in the pocket. I'll pass it around. You can also buy it. It costs about $4 or $5, I think, something like that. Between $3 and $5. And it has 6 parts. Let me just pass this one around. And after the break, maybe I'll bring all the copies so that you can see the different parts, and we'll talk a little more detail about it.
Pope's encyclical is on, you can get it through Amazon also.
Yes?
For $12.95, free one-day delivery. You can also go right to people's Kindle.
Oh,
if you want.
yeah. You can download it to the Kindle. You can
Amazon has it in a paperback and on a Kindle format.
Super. You can also download it to iBooks if you want for free, again from the Vatican site. Okay. You can download it on your, if you have a pad or the phone. So the Vatican now is trying to put all their documents for free online. And the advantage of that also is that it's in different languages. Okay, so that's basically the textbook for this course. We're into part 4 here, part 4, which is issues in care for the beginning of life. Obviously, this is for the beginning of human life because it's on healthcare. Okay. So it's only about 2 or 3 pages long, that section, but it's very succinct. It has specific directives. Okay, let me get into the presentation if there's no more questions or comments. We left off, I was talking about the 2 basic modes of reproduction last time, which are universal, okay? Meaning that whatever species we have observed, we have seen that there are these 2 modes of reproduction, which again, staying with the big picture for now, remember that reproduction is one of the 2 basic characteristics for being alive, or being alive organically. One is metabolism, which is another way of saying body functioning or being alive, necessary but not sufficient. That organism also needs to be able to generate another one like itself, the next generation, right? The offspring. And that, again, we see that these are universal things that happen in nature. Every species, in order to continue, needs to reproduce. So then we see that there are these 2 modes, the asexual reproduction that I talked about last time, and now sexual reproduction, which is the one that I'm concentrating on because it's how we humans reproduce. So I'm also going to get a little bit into early embryonic development in some level of detail. And then if we have time, the ontological status of the human embryo. Ontological meaning the status of the human embryo as such, as a being, as a living being. Who or what is the human embryo? Okay. So, sexual reproduction involves fertilization. Fertilization involves the fusion of gametes, right? Gametes are the sex cells, and the gametes are what we call haploid. In other words, they don't have paired chromosomes. They're unpaired, so that when fertilization occurs, then the diploid number of chromosomes is restored. All right, so it's a very, it's a very basic, simple arithmetic process that is going on. And yet, how many people get confused with this process, which is called meiosis, to get to that, to reduce the number of chromosomes so that at fertilization, that number may be restored again to what we call the diploid number. The diploid number, I talked about this last time, the diploid number is represented algebraically, if you will, as 2n. The n stands for number of whatever species, because this applies, again, universally. The vast majority of organisms in nature have diploid cells. Their bodies are made up of diploid cells, right? And so numerically, diploid is represented this way. The N, again, is the number of chromosomes unpaired, the single number of chromosomes. So for the human, how much would N be? The unpaired number is 23. You see, the unpaired number is 23. So multiplying 23 times 2, that's where we get the total number or the paired number of chromosomes, right? Which is 46. And that's what we call the diploid number. So the diploid number of chromosomes in the human, 46. That's a straightforward question that can be asked to any species. And also it helps because it starts defining biologically, organically, what species the individual belongs to. It's not just about the numbers, though, it's also about the qualities, not just the quantity, but also the quality of chromosomes, all right? Because there are some species that may have also 46 chromosomes or 23 pairs, but they don't have human chromosomes. So they would be another species. In general, the larger the number, the more sophisticated the individual, or the species, but not necessarily. For example, chimpanzees, their end is 24. So they have 48 chromosomes, or 24 unpaired, yeah. And troglodytes is 24 pairs of chromosomes. So they have one more.
Chromosomes, do they know why?
No, it's just the way that chromatin actually fragments and splits up to make the chromosomes. So also keep in mind that chromosomes are just you can think of it like a bus. You know, the bus can be full of students, full of people, or empty, or half full, or quarter full. What the chromosomes carry is the genes. We have between 10 to 20,000 genes to make up the species, and those 10 to 20,000 genes are spread out among the 46 or 23 pairs of chromosomes, all right? And it's not necessarily the same genes that are on the same chromosome for the different species. That will vary, okay? And in general, the larger the chromosome, the more genes on it, but that's just general, not always. Talk a little bit more about chromosomes later when we look especially at the sex chromosomes, which are the ones that determine the sex of the offspring. All right, so we can say that as far as genetics is concerned, for the genetics of it, the fertilization process essentially is to restore the diploid number of chromosomes in the offspring, in the new individual, okay? To restore the diploid number. In other words, so that this new individual will have a backup set of chromosomes, just like the parents have a backup set of chromosomes, all right? Just like mom and dad have a backup set of chromosomes, you know, the diploid cells, the offspring will be the same that way. So again, just for nomenclature, this is, so that we're all on the same page. I know that a lot of this is just old stuff, but basically the gametes are the sex cells, all right? Which are sperm or egg, and they are produced by a process called meiosis. Meiosis is like a double mitosis, and you'll see what I mean. It's like a double mitosis, and that's where the number of chromosomes is reduced to the unpaired number, Okay, so let's get into, first let's do a little review of mitosis so that you see how the chromosome number is maintained in mitosis, which is just straight up cell division, okay? We start with a cell in interphase, which in interphase, the cell is metabolizing, right? The cell is just doing its function, whatever function it is, whether it's a liver cell or a brain cell or a skin cell, they're just doing their function, they're specialized. And the DNA is in the form of chromatin or shape of chromatin, which is a single string. You can think of a ball of spaghetti with a single string of spaghetti in it. And it's a little, that structure is a little more open than the chromosome because it's being transcribed, right? It's being transcribed so that then the messenger RNA can be translated into proteins that do the actual functioning, the actual metabolism that is occurring. So really, when you think about it, it's all very mechanical. It's amazingly mechanical. It's chemical and mechanical in the same way. But if you look at the chemical aspect, in other words, the molecules they have a three-dimensional shape. And it's all about that three-dimensional shape that makes the whole thing work. All right, now when the cell is going to divide, the metabolism is shut down to the minimum. And it's just the metabolism that divides the cell. Okay, so the cell is going to concentrate all its effort into this division, into this splitting into two. And two major things need to happen. The first major thing is that the chromatin needs to duplicate. All right, so if we started with a 2N cell, temporarily going into prophase, it's going to become temporarily 4N. And look at this. So we can ask, how many chromosomes in this cell? I hope you all agree there are 4 chromosomes. 1, 2, 3, 4. 2 pink and 2 green chromosomes. You all agree on that? There are 4 chromosomes, okay? They look like little X's. I'll get to that in a minute. But basically, there are 4 chromosomes here. Also, the way the thing was drawn, there are supposed to be 2 large chromosomes and 2 small chromosomes, which are supposed to be the same size. So this pink chromosome is the same size as this green chromosome, and this little one is the same size as this one here, all right? What does that mean? I think I labeled it here, relabeled it. Yeah, okay, so we got chromosome 1A and 1B and chromosome 2A and 2B. Those are the pairs. So I can ask, how many chromosomes in this cell? In prophase? 4 chromosomes. How many pairs? 2 pairs. The small pair and the large pair. Okay? Stay with me. Now, each chromosome, each one of the 4 chromosomes has already duplicated because it has the sister chromatids, you see? So we can say that this is one side of chromosome 1B, and this is the other side of chromosome 1B, which is identical. So the duplication, the replication has already occurred here, all right? Has already occurred. So technically, when we count here, we have 4 chromosomes that have already replicated, but they have not separated because there's a little structure that is holding them together. At the middle is that white dot that is called a centromere, all right? And so the centromere is holding the duplicates together. So we can say that technically this cell actually is 4N. You see, because the number of chromosomes is 4, or sorry, the single number is 2, that's a single number. Well, we got each chromosome is double, so that's 2 times 2 is 4, and there are 4 of them. So this cell temporarily is a 4N cell, simply because the number of chromosomes has already replicated or duplicated. I say it's temporary because when the cell continues to split, then the chromatids, the sister chromatids, are going to separate. In other words, the centromere is going to give way, and the sister chromatids are going to separate. And that's essential so that the daughter cells, the 2 daughter cells that result, each one has 4 chromosomes, just like the original mother cell, okay? And there's no loss of chromosomes. Imagine if there was no replication. You're all familiar with replication or duplication because you had it in the summary, all right? Imagine if the chromatin did not replicate before dividing. And then we proceed with the division from 1 cell to 2 cells. What would that do to the number of chromosomes? It would split it in half. So that if this cell had 4 chromosomes to start, each daughter cell would have only 2 chromosomes, okay? And so for that not to happen, for the daughter cells to continue to be diploid, to be 2N, a duplication has to happen just before the split, okay? All right, then comes the split, and these are the 4 phases. I'm not interested for you to know the actual names of the 4 phases, but that's what they are, that's what they're called. In the first phase, the chromosomes are already duplicated, all right, but still together, held by the centromere. Then those chromosomes line up at what we call the equator of the cell, which is the middle, the equator of the cell. And then in the next phase, the individual chromatids, so for example, this chromosome has 2 chromatids, okay? And this one has 2 chromatids, this one has 2, and this one has 2 chromatids. The individual chromatids are pulled to opposite parts of the cell, to the opposite poles. So this is called a pole, and this is another pole, all right? So the sister chromatids are pulled to opposite sides of the cell, which is still a single cell, but undergoing mitosis, undergoing division. And so the centromere gives way. This is, again, a very mechanical process. There are these spindle fibers, which are mostly proteins that come down and they pull. They're like coils that pull on opposite sides of the chromatids to split them to go to their place. So I'm saying this is a biochemical process that is a very mechanical thing. It's like having 2 hot dogs next to each other and tie a string to each one and pull in the opposite direction, all right? That's a kind of a crude way of telling you what is happening mechanically in the cell. But it does happen, and it's at the molecular level. So it's just amazing. We call this thing elegant. All right. Finally, once the sister chromatids have reached the poles, the opposite poles, then a nuclear membrane is resynthesized. Right? Nuclear membrane is resynthesized around each one of those clusters of chromosomes. And then finally, the last stage is the pinching of the cytoplasm in the middle. Just pinches as if you were to take a balloon, pinch it in the middle, or kind of twist it, right? That will pinch in the middle and then seal off, and we'd end up eventually with the 2 daughter cells. This part of the process is called the cytokinesis, all right, which is the actual pinching. But already the 2 nuclei are intact with the full number of chromosomes each, with the 2n number. So that's straight up mitosis. It involves, like I said, 2 steps, or the first step is a duplication of the chromosomes, of the chromatin, going into chromosomes. And then the second step is migrating to the opposite poles so that the 2 daughter cells will end up with the same number as the original mother cell. And I think here I go through stages. I just labeled it, yeah. This one in the interphase, here is the replication from a single chromosome to the sister chromatids. So here we have actually 2 identical chromosomes, but they're held together by the centromere, all right? They're actually called alleles. So that's the DNA replication process here, and these are the sister chromatids. That happens within this region of the cell cycle. If the cell undergoes a cycle like this, the first region is called G1. The first part of the cycle is called G1, the second S, the third is G2, and then here, finally, after G2, the cell undergoes reproduction or mitosis. For the sake of completion, G stands for gap, or you can also see it as growth, the first part of the growth of the cell. Then the S stands for synthesis, which is another word for replication. So DNA duplication, DNA replication, DNA synthesis, it's all the same thing. It's duplicating the darn chromosome, okay? So here you can see you have a single pair, now you got the double pair. The gap 2 is the second part after synthesis. There are checkpoints here that the cell has to go through to make sure that the chromosomes have duplicated before the cell goes into gap 2. So after it goes into gap 2, then gets all the okays, all the biochemical signaling ready to go, and then the cell starts the splitting process, lining up at the equator, migrating to the poles, pinching at the middle, 2 daughter cells. So that's the whole process in detail. Now, any questions about mitosis? Stay with the big picture. One mother cell, 2n. At the end of the process, 2 daughter cells, each with 2n, because those chromosomes duplicated or replicated before the split. All right? Okay. Meiosis. one way of looking at meiosis is like a double mitosis with a twist, okay? So it's a double cell split. So from one cell, one mother cell, we're gonna end up with 4 daughter cells. Or if you want, 2 daughter cells and 4 granddaughter cells, okay? Either way, but the point is that after meiosis is complete, we end up with 4 daughter cells or 4 cells from the original 1 cell. Another thing that occurs during meiosis is this process of recombination, which is also known as crossing over. I'll show you what that means. And the most important thing for meiosis is that it reduces the number of chromosomes from diploid to haploid. Haploid meaning single. Ha is Greek or Latin for single or 1. And so we go from 2n to 1n. But typically when there's a 1, as the coefficient is not shown. So when you see, it's just like in algebra. A plus B, well, it implies that there's 1A and 1B, all right? So when the coefficient is 1, it's not shown, and therefore we just put n for the haploid number. So that is known scientifically, chromatic reduction, again, because we don't want people understanding what we're talking about, so we call it chromatic reduction. It means that the number of chromosomes is being reduced typically to half. Or the unpaired number, which is more informative. It's the unpaired number of chromosomes. So this is representing a human cell. In mitosis, the mother has 46 chromosomes and the 2 daughters each have 46 chromosomes. In meiosis, the mother cell has 46, but then through the double split, each one of the 4 daughter cells is going to end up with only 23 chromosomes, the unpaired number.
Is the second step in
Yeah.
meiosis,
Yeah.
the middle one,
Here?
is that an actual occurrence or is that?
Yes. No, it's an actual occurrence.
So
It's
what's
a
the
double
number
split.
in that- What's the number in those 2?
Good, thank you. This is still 46.
Okay.
So the chromatic reduction occurs in the second meiosis. Okay, yeah. This is still, they're still diploid, but they're in the process of becoming haploid. So the chromatic reduction occurs in the double split. And that's why, basically, because what happens here, at this level, there's no replication again. This cell only has, it's locked in. This cell only has 46 chromosomes, has the pairs, but it's not going to replicate again. It's not going to duplicate. So when this cell splits, then it has to share the pair, okay, and unpair. Mm-hmm.
So let me see if I have this right. I'm going to use my big vocabulary.
Yeah.
The mitosis
Mm-hmm.
occurs in somatic cells.
Exactly.
And the meiosis occurs in gametes.
Yeah, exactly. That's exactly right.
They never are.
That's right.
It's always mitosis for somatic cells.
Correct.
Always meiosis.
By definition, because somatic cells is another word for body cells. They typically, the body cells, the somatic cells are specialized. The vast majority of cells in our body are somatic cells, and they are specialized or differentiated, fully differentiated, right? And they're doing their function. Again, it's according to the tissue of the organ where they're working. And so specialized or fully differentiated cells, they are somatic and they are diploid, and so they are undergoing mitosis. That's for organ regeneration and tissue and so forth. Meiosis, then, these cells already, these are special cells. These are the primordial, or these are the precursors of the gametes. And these cells, even though themselves are diploid, they are locked in to go into producing gametes. Therefore, these cells are only found in ovaries and testicles. And these are the cells that are in the follicle of the ovary, and these are the cells that are the precursors in the testicles to form sperm. And those are the specialized organs for reproduction. So again, biologically, genetically, the purpose of the sexual organs is to house diploid cells that are specialized into undergoing meiosis instead of mitosis. And that's how we get the gametes. Okay, a little bit of detail here just to track the chromosomes. The cell in interphase will duplicate, will replicate, all right? And so here's, meiosis then is divided into meiosis 1 and meiosis 2. For the first split is meiosis 1, and for the second split is meiosis 2. So pretty straightforward, all right? Meiosis 1, here you see how many chromosomes in this cell? How many chromosomes? 4 chromosomes. How many pairs? 2, assuming that the large ones are the same size and the small ones are the same size. So 4 or 2 pairs. All right, diploid cell, right? Because they're paired chromosomes. How much is N in this diploid cell? Not Luis. Anyone else but Luis. How much is N here in this cell? See where I'm at? Yeah, 2. 1 and 2. There are pairs. It happens to be 2. They could have put 6 chromosomes in here. If there were 6 chromosomes in here, how much would N be? N would be 3, all right? Because they're paired, assuming that they're pairs, okay? So if they have put, let's say this is a small and this is a large, let's say that they have put 2 fibers in here that are intermediate between the small and the large, but those intermediates are the same size, they're both the same size. That would have been chromosome 1, 2, 3, or actually 1 is the largest, 2 middle, and 3 would be the smallest. You would have 6 chromosomes total. The 2n number, the n for that 2n would be 3, because 3 times 2 is the 6. Okay? If we had 23 pairs of chromosomes in here, let's make this a human cell. Let's say that we had 46 chromosomes in here, and each one of those 46 was a pair, all right? How much would n be? 23. Don't be confused by the address. So I think that the brain does math in a particular area that is not used all the time. So when it's used, people freak out. The brain freaks out when, oh no, I have to use this area. I haven't gone into that dark room for ages. And now the professor is forcing me to go into that dark room. I don't know what I'm going to find there. He's asking questions. Right? Okay, so it's very straightforward. Here you have, in meiosis 1, the 2 pairs of chromosomes with their sister chromatids. Okay, so this is the temporary 4n stage, right? The difference in meiosis from mitosis is this. You see how the chromosomes are lined up here at the equator? They're lined up singly. Singly meaning here's chromosome 1a, here's chromosome 1b, 2a, and 2b. But they line up singly. Each chromosome has its sister chromatid, yes. But as a whole chromosome, they line up singly. All right? Look at this. Here, in meiosis 1, when the chromosomes line up at the equator, they line up in pairs. They line up paired. All right? So there's a level of organization there in these cells that does not occur in mitosis in somatic cells. This is the gametic precursors, all right? The germ cells for gametic precursors. And the lining up of these cells, of these chromosomes, is in pairs. So that when they migrate, when the chromosomes migrate to the opposite poles, the sister chromatids don't split. They stay together. That's the trick here. That's the key. The sister chromatids do not split. So the machinery, the spindle fibers, they don't pull they don't disrupt the centromere. They leave the centromere intact so that the pairs migrate to opposite poles. So again, if we were to label this, this would be 1A, this would be 1B, 2A, and 2B. So the sister chromatids migrate intact to each pole. That's the key here to eventually ending up with 4 haploids. Because what happens? When meiosis 1 is completed, and you look at it, actually each daughter cell is like the mother cell. Each daughter cell is like the mother cell. Okay? So at the end of meiosis 1 going into meiosis 2, each daughter cell is like the mother cell. Look at this. Here, if we show this diploid cell after replication, it would look like this. Okay? Now, what happens? In meiosis 2, these chromosomes do not replicate again. The cell will divide again, but there will not be a second replication. That means that when these chromosomes line up at the equator, now this one is like an straight up mitosis, basically. When they line up at the equator, now the sister chromatids will separate at the centromere. At the centromere, the sister chromatids will separate, so that in the end, here, we'll end up with single number of chromosomes, not pairs. So there was a replication, a single replication at the beginning, going into meiosis 1. But going into meiosis 2, there was no replication again. And that's why the end result is that the chromosomes are unpaired. But there's a double number of cells, because there's a second split, and necessarily when you take 2 cells and you split each one, you end up with 4 cells. So here are the 4 cells represented already, fully split, okay? And you notice that these 4 daughter cells have unpaired chromosomes. For example, the large blue, and then on this one. And here, but here it has the red, the small red, whereas these other 2, they have the large red, but the small blue, okay? So you notice that there are no pairs here. It's only one large and one small on each one of the 4 cells. And therefore, these cells are haploid, represented as N, and these are the gametes. Again, for mammals and for the vast majority of species, in the female, these are eggs, and in the male, these are sperm cells. So again, the big picture, I'm not concerned about you knowing all the stages of meiosis 1 and meiosis 2, but I want you to know that because this has an implication for ovulation, the first ovulation menarche, okay? Which in women, they're born with a set number of eggs, and baby girl in arrested stage of meiosis 1. And it takes another dozen years on average, more or less, for meiosis 2 to complete. And finally, she'll produce a first mature egg, and that's the menarche, that's her first ovulation. Right? So there's an arrest that goes here, biochemically, for about a dozen years on average, 10 years, whenever you want to put menarche. Different from the male. We'll talk about that. All right, so there's another thing, another detail here. The reason for fertilization, the reason for, sexual reproduction. The intuition should be that if a cell has to go through all this, energetically, it's more expensive. It takes more energy to do this for each cell. And in fact, it takes, on average, twice as much energy. So if it took, I don't know, 1,000 kilocalories to do one mitosis, I'm just pulling a number from the hat. Let's say if energetically it took the cell 1,000 kilocalories to do this. To do this, it will take 2,000 kilocalories. So it's not as economic. There has to be a justification, otherwise selection, evolution would have killed it. You know, it's too expensive. It's like having to push the car up the hill all the time. At some point, you just give up with that darn car. You give it up, okay? Get a car that has a motor. So that's the point. It's about twice as expensive energetically for a cell to undergo meiosis than to undergo mitosis. So what's the kickback? What's the advantage of doing this? The advantage is variation. Remember that selection selects on variation. As long as the climate doesn't change, variants are no different, right? They don't have an advantage. But when the climate changes, some variants on average will have an advantage over other variants. The ones that are taller will have an advantage, or the ones that are smaller have an advantage, or the ones that are more resistant to UV or more have an advantage, or the ones that don't have as much resistance to UV. You see, so variation translates into potential for survival, for better survival. Where's the variation? The variation is in the process called recombination or crossing over. I don't know if you can see it on the screen, but if you notice here, these little chromosomes, you notice how they have the red, the blue has a little red point at the bottom, and this red one has a little blue point at the bottom, okay? Here, I don't know where you can see it best. See this red chromosome has a little piece of blue underneath, whereas this blue has a little piece of red. How did that happen? They crossed over here precisely when they line up in pairs. When the chromosomes line up in pair in meiosis 1 here, the equator, before splitting, they interchange pieces of chromosome or pieces of chromatids, alleles, all right? This is the crossing over event. Let me show it to you in a little more detail. So here's how many chromosomes here? 2 chromosomes. How many chromosome pairs? 1, they pretty much look the same size, right? Okay, how many chromatids, how many sister chromatids in this chromosome? 2, right? Here's 1 chromatid, here's the other sister chromatid. So has this chromosome undergone replication? Yes, because you got 2 sister chromatids, okay? So this is almost identical to this, okay? And the pair also underwent replication. So this chromatid is almost identical to this one. I'm saying almost because it could have some mutations on it. But essentially, all of these 4 sister chromatids are pretty much the same. They have the same information, more or less. Okay? But one can have dominant genes and the other one can have recessive genes at the level of the individual genes. So what happens? During meiosis 1, when these guys line up in pairs, okay, not singly, but in pairs, they like each other so much that they start sharing pieces of the hot dog, okay? So the blue one, I like the red hot dog, so I'm gonna take a bite of the red hot dog and keep it with me. And then the red one likes the blue hot dog, so it's gonna take a bite of the blue and keep it with me. So there's this event of crossing over or recombination. This has been studied in detail down to the molecular level. It's all regulated by enzymes and by machinery that does the splitting, the nicking, and then the grafting, and then the sealing, and all that is done in a very elegant way, again, at the biochemical level. The end result is that the chromatids that exchange genes, that interchange genes, okay? Those are called recombinant chromatids because they recombined. And the other chromatids that did not exchange, they're non-recombinant because they did not exchange genes, all right? A little too technical, don't worry about the words. I'm just telling you, for the sake of completion, what it's called. But the event itself is called recombination. And this allows for variation. Because at the end of the day, whatever gamete, whatever egg or sperm, see, each one of these is going to end up in a different egg or sperm. This chromosome is going to end up in one egg. This chromosome is going to end up in another egg of the 4, okay? And so, you see there's a recombination here. This chromosome has all the genes identical to this one, except this tail end, which has the genes from here. All right? Now, they're the same type of gene. Let's say they code for the color of the eye. But maybe this one codes for blue eye, and this one codes for brown eye. So it means that this blue chromosome is going to code for brown eyes, and this brown chromosome is going to code for blue eyes. The region codes for eyes, but the color is different. It could be it could be that centuries from now, there would be a selective advantage for having blue eyes or for having brown eyes. The recombinant is going to have this selective advantage. Okay? So anyway, this issue of recombination or crossing over is what allows for variation. And that's why we don't look identical to our parents. I don't look identical to my father, and the daughters don't look identical to their mothers because of the variation that occur. All right, that's one reason. The other reason is because precisely the egg and the sperm, which come from two individuals, are fusing into a single new individual. So even though we share human genes at the level of details, the liver could be a little larger, a little smaller, more functional, or less functional, okay, at the level of detail.
And this happens every single time.
Every single time. Recombination is the big advantage of sexual reproduction, of meiosis. And it happens because the chromosomes line up in pairs at the equator instead of singly. This is more representative, this diagram. You see, because here they're lined up singly, therefore they cannot exchange anything because they're next to each other like this in a series. But here they're parallel. So because they're near each other, hey, quickly, let's exchange some information here before they split us. Okay? And again, this is all done chemically. We don't have to worry about it. We don't have to tell the sperm, okay, recombine, please. No, it does it automatically, and that's what we call it elegant. Okay, because this has been going on in evolution for billions of years, and it's a, you know, it works, and therefore nature doesn't fix it. It survives the selecting out process. It survives selecting out. Okay, it introduces recombination. Therefore, after mitosis, the daughter cells are essentially clones of the mother cell. But in gametogenesis, in meiosis, the daughter cells are not identical clones. They're little recombinants, okay? Little recombinants. So just keep in mind that introduces variation. Okay, so, oh yeah, here I lined it up, the diagram of recombination with the diagram of the actual lining up at the equator and then splitting. So it's a little more illustrative, but this is where it occurs, okay? These guys, here is a recombination event, so that when the splitting occurs, then you get the recombinant alleles or the recombinant chromatids, together with the non-recombinant system. Meiosis 1. By the time meiosis 2, what happens? The centromere splits, right? Centromere splits. So whoever recombined, and whoever didn't recombine, didn't. So from here, for example, from this pair, we get 4 daughter cells. One daughter cell is going to have full blue. The other daughter cell is going to have blue-red. The other daughter cell is going to have red-blue. And the other daughter cell is going to have red. You see? Because the sister chromatids then in meiosis 2 will split at the centromere and will go singly to each pole. We can say then, following this argument just one step further for a moment, the 4 daughter cells that result from this chromosome, 2 are recombinant cells and 2 are non-recombinant cells. In other words, the 2 that are non-recombinant, those genes in there are identical to the parent, to the mother gene. But the other 2 are recombinant. And there's a mixture, typically, like I say, of dominant or recessive genes, all right? Dominant or recessive.
Is that a law? That's something that repeats itself, or is that an approximation? Like, we always have 2 recombinant and 2 kind of pure conserved.
Okay, the short answer is I don't know, but my suspicion is that, so this mechanism is so elegant, as you know, down to the molecular level, all right, that it could be that this chromosome actually recombines at another tip also. Or it could be that this tip recombines with this one. You know, if we label them chromatids 1, 2, 3, 4, it could be that at the long arm, at the P arm, 2 and 3 are recombining, and at the Q arm, 1 and 4 are recombining. Okay? I don't know, but I'm sure that there is a mechanism that regulates all that, the recombination. So you have many different possibilities, really, but the point is that it's equivalent genes that are being exchanged. Okay, always equivalent genes. Not one gene is switched for another different gene. All right, backing out to the big picture then, and some little more nomenclature here. Meiosis, formation of gametes or sex cells, and they are haploid, unpaired chromosome numbers, okay? And that's why it can be an even number or an odd number, it doesn't matter, but they're haploid. Diploid number is always an even number because it's 2 times something. All right, so because the formation of gametes is called gametogenesis. Genesis, again, a reference to origin or beginning or formation. Formation of gametes is called gametogenesis. If it's for sperm, it's spermatogenesis. If it's for eggs, it's oogenesis. All right? And again, haploid illustrated by the single number. Moving forward. Now, let's look for a moment at eggs and sperm in comparison. So here are the spermatocytes, which are diploid, which are in the testicles, inside the vas deferens of the tubules inside the actual testicle. Okay, and these are primary cells, they're diploid, but they are programmed to undergo meiosis. Okay, and the same case, the complementary case in women, the oocytes, the primary oocytes, again, they're diploid. They're inside the ovaries, which eventually will become the follicle. So they go their replication, they line up at the equator, and then they migrate as entire chromosomes without splitting the centromere to either side. Okay, so far the process is analogous for egg and sperm until we get to meiosis 2. Actually, already in meiosis 1, there's a difference. Notice, it's kind of the detail, notice that at the end of meiosis 1, the 2 daughter cells have the same size. So they get equal amount of cytoplasm from the primary and secondary spermatocyte, okay? So at the end of meiosis 1, the spermatocyte cells are identical in size. They get an even amount of cytoplasm. So basically the cell pinches at the middle. Unlike eggs, ova, what happens after the splitting of the primary oocyte, at the end of meiosis 1, there's an unequal distribution of cytoplasm. The chromosomes split equally and so forth, all right? So that there's 2 chromosomes here and 2 chromosomes here with the sister chromatids intact. But this cell has more cytoplasm than this cell. And the same happens in the second split. So that the cell that got less cytoplasm doesn't have enough cytoplasm to survive. And even this one is overrepresented in size. It should have been even smaller. Because at the end of the day, these 3 become the polar bodies, which are non-functional. Okay, and they're degenerate, if you will. But one of the 4 got the bulk of the cytoplasm, which is where metabolism is occurring, which is what is keeping the cell alive. And so that's why, at the end of the day, functionally, for every follicle that matures, we get 1 egg, but there are 3 little polar bodies that are hanging around, and typically they're encrusted on the membrane, outside the membrane of the cell, because it's not just an egg like that, okay? No, it's a whole structure, it's a whole cluster. It has the zona pellucida, the corona radiata, so it's a whole cluster of cells. And those 2 layers, the zona and the corona, are protecting the egg because the egg is super delicate. It's unpaired chromosomes, all right? And therefore, any damage could become a mutation, which could be passed on to the next generation, which is not a good thing. So the egg is protected by the corona and the zona, but inside those layers are 3 little polar bodies that are the degenerate eggs, if you will, that did not get enough cytoplasm. All right? Unlike with sperm, for every primary spermatocyte, we get 4 sperm. And there are millions of spermatocytes, and that's why we get gazillion sperm. In fact, for an ejaculate to be considered functionally potent, capable of fertilizing, you know how many sperm it needs to have? Clinically, for a human ejaculate to be considered fertile, competent, capable of fertilizing, about 100 million sperm. 100 million sperm. That's called healthy. All right?
I've heard that, well, it is increasing significantly on average on the whole population.
Really?
At least in the developed world.
Yeah. Interesting. It'd be interesting to see why. Could be a combination of factors. Typically, some kind of lifestyle that is affecting, right? Could be the food that we eat, could be radiation, who knows? But yeah. Part of it is, yeah, we'll get to that. We'll get to that fertility factors. But that was my train of thought here. Yes, 100 million for competency. So that's called sperm competency, or you hear about incompetence, sperm incompetence, which, by the way, is a minor. Traditionally or historically, when there was infertility in a couple, it was automatically the woman's fault, right? Because you're always at fault. I mean, no matter what happens, you're always at fault. So automatically, the woman was at fault because why? Well, because here's the semen. We can see the semen. So obviously the man is competent and he can get an erection. So he's okay. It's the woman's fault. And that historically in the Jewish tradition and in other traditions was automatic grounds for dismissal and divorce. Okay, because she was infertile. And it was all about having the children, the progeny, and the inheritance, and the chosen people, and all that. So automatically it was a decree of divorce, and it was a big shame factor, and woe is me because I am infertile. I go into the desert to mourn my infertility until God kills me, and so forth. So it was a big, big taboo. It's been determined today that infertility now is about half and half. Half of the time it's on the woman and the other half of the time it's on the man. Or sometimes it's on both, all right? But so that was a horrible myth that was placed on women automatically, simply because visually they could see the semen and so forth, so they just figured automatically it was the woman's fault. A lot of misconceptions there by ignorance and arrogance, I would say, but that's how it is. Okay, let's move forward a little bit here before I take the break, which will go at the process of fertilization. All right, so again, this is kind of a big picture slide for our species, number of chromosomes, total number, 46, or 23 pairs. But technically, the 23 pairs only happens in the female with XX, because for males, the sex chromosome is XY, which is not a real homologous pair. Okay, I mean, it is a pair, but they're not homologous, they're not identical, because obviously an X is not the same as a Y chromosome. And the Y chromosome, oh, that's what I had. The Y chromosome is also getting smaller and smaller. So, yeah, the Y chromosome is getting smaller. So there could be a time that the male disappears. We'll see, that's an interesting scenario. We all become parthenogenic. Yeah, but the Y chromosome over decades and decades, over generations, is getting smaller. It keeps losing. There's very little information on the Y. It's mostly the SRI gene, and a few other genes that determine maleness. But it's a real degenerate gene, literally, biochemically speaking. All right, so we got the pairs. Of the 23 pairs, 22 of them are called autosomes. They have nothing to do with sex. They have nothing to do with the determination of sex, all right? They code for just the gazillion other characteristics of the human. The fact that we have a head, where the head is placed, where the arms come, the fact that we have 5 fingers at the end of each arm, etc. All that coding is in the autosomes, in the non-sex chromosomes, okay? The one pair that codes for the sex, for determining literally, biologically, physiologically the sex, in other words, either male or female sexual organs, including primary and secondary sexual characteristics, that's known as the sex chromosomes. That's the one pair, all right? So humans, N is 23 chromosomes, only in the gametes, and 2N, 46 chromosomes in these somatic cells or body cells that are doing every other function but reproduction. Okay, diagram of the two processes in situ in the actual organ. Here is the ovary, here is the testicle. So for the ovary, we have the follicles. As the follicle matures, primary oocyte, remember, is still diploid. Is that too small? Can you see? 46XX? Can you see it, or is that small? Can you see it in the back? 46XX? No? Let me make it a little bigger. Concentrate on one. Okay, good. Now, this is genetic nomenclature. I don't want you to get confused, all right? It's just genetic nomenclature. And the genetic nomenclature is this. See, 46 is the total number of chromosomes. It's the diploid number of chromosomes. And XX is just showing, it's illustrating the sex chromosomes. That for this cell, the sex chromosomes are XX. Don't interpret this as 48, 46 plus 2. No, 46 is the total number. The comma is significant. It means that it's only showing one of the chromosome pairs, which is the sex chromosome, to determine the sex of this cell, all right? So the same argument goes over here. What's the total number of chromosomes? 46, all right? And in this case, the sex chromosome is XY, the sex pair. But it doesn't mean 46 plus 2. No, these two are already embedded in here, all right? It's just a nomenclature. So, and it determines that it's just illustrating that this particular cell is a male cell, and therefore it's a it's a primary spermatocyte and not a primary oocyte. All right, then we go down to the split, but you notice that this is being done functionally, meaning that it's not showing the polar bodies. Okay, so, but it is showing the follicle. So you see differences as the spermatocytes are splitting. This is the meiotic division, first meiotic division, second meiotic division. In result, 4 sperm, each one with a haploid number, 23. Half of the sperm is going to be X and the other half is going to be Y. So we can say 50% now, looking at it functionally, let's say with spermatogenesis for now, okay? 50% of the sperm on any ejaculate, on average, is going to be X sperm, and 50% is going to be Y sperm. Does this mean that this is female sperm? No, it's male sperm because it comes from a male, okay? So it's sperm, it's not an egg, but it has an X chromosome, whereas this sperm has a Y chromosome. So when fertilization occurs, who determines of the two cells, of the egg or the sperm, who determines the sex?
The male.
The sperm determines, because if it's an X sperm that fertilizes, that baby is going to be a baby girl. If it's the Y sperm that fertilizes, the baby is going to be a boy, okay? And so again, very straight up, depending on whether the sperm has X or Y, that determines the sex. I'm talking about normal, on average. There's always variations from this. There could be a wrong split of the chromosomes, and it could be that this, let's say, for example, at this level, if this chromosome gets damaged, then these don't get, these sperm don't get an X chromosome. Or it could happen here also. When the fertilization occurs, this could be an XO fertilization, or X0 didn't get a second sex chromosome from the sperm, or vice versa. Those are abnormalities, thanks be to God, that occur very rarely, and typically they are infertile, so it's a dead end. Genetically, it's a dead end phylogenetically. It doesn't get passed on because they're infertile. They don't produce fertile organs, basically. They don't produce fertile sexual organs. So whereas those abnormal folks can live, like the syndromes, like Klinefelter syndrome, Turner syndrome, and all those, they can live their lifetime. They sometimes have a biggest genitalia, but typically they are infertile. So they will not pass on the anomaly that they received, okay? But for the vast majority of the rest of the population, it's the norm, this is the standard. So functionally, from one primary spermatocyte, we get 4 sperms, half of which are X sperm and the other half are Y sperm, all right. For the egg, functionally, it's a little different, genetically the same split of the chromosomes and so forth, But you notice what is not illustrated here is the polar body. That would have been here, one branch of a polar body that is degenerate, actually stays embedded in here somewhere. And then here again, another uneven distribution of cytoplasm, so another polar body, okay? They're not shown, but what is shown is that the follicle stays with the egg. So this is the egg inside here, okay? But you notice that the egg has layers around it, basically two layers. The one layer is called zona pellucida, and the next layer is called corona radiata. The corona radiata is actually a cluster of cells or a cumulus of cells. And so the egg, functionally, it's an entire package. And the sperm needs to penetrate all that in order to release its nucleus inside the egg, right? And so it's protection, and again, all that is done in a biochemical process. Let me get back up here for a moment. So you see the follicle, then when it ruptures, the egg ruptures, it doesn't rupture just its single egg, just one cell. It carries with it a cluster of cells and layers that form those two layers that I mentioned here, illustrated in the blue, light blue and dark blue. Okay. All right. Gonna stop here for a moment. I've given you a lot of stuff. When we come back, we'll watch a little video on fertilization at the cellular level, and then we go from there into the actual fertilization process and show you some more stuff. And briefly, we're gonna go over embryonic development. Okay, so it's 11 o'clock. Let's take about 10 minutes, please, and we'll be back. Let me ask any questions or comments. No? All right, so about 10 minutes and we'll be back. Let me pause recording here. Okay, so welcome back. Now we're going to go into fertilization and embryonic development a little more quickly. I wasn't planning on taking so long for the first part, but you want to close that, please? Thanks. Here's some people.
Yeah, they're
always
visitors, I guess.
coming around with showing students. Okay, so this little video I found interesting because it has, it's a couple, it seems to be a French couple, and he's very savvy with technology and created this, this video, and his wife is pregnant, and at the very end, they show us the baby. But he's done partly graphics and partly photograph or filming, but it shows further fertilization process from the moment of ejaculation at the cellular level, how the sperm is traveling through the whole duct of the fallopian tubes until it reaches the egg, fertilizes the egg, and then the egg, which is now an early embryo, starts rolling down the fallopian tube and falls into the endometrium, embeds itself. It's just fascinating. But I want to point out at one point, there is a hatching process. Now, you never heard of mammals hatching, right? We're used to birds and fish, reptiles hatching, but there's also a hatching process for the human, for mammals, when the little embryo leaves behind the cumulus of the zona and the corona radiata, all right? And literally, the little embryo has to crawl out before embedding into the uterus. The other thing is, for some reason, this fellow was into acid rock, and there's no narrative to the film, but he's got acid rock music in the background, guitar, which I totally, absolutely hate. So I'm gonna block the sound and just do my own narrative on it, okay?
We have to listen to the sound at home.
You're welcome to it. Just don't send it to me.
We'll tell where we got it.
Right. Or you can just play the sound part if you like. Yeah, he calls himself Evil Cat for some reason, but let me block out the sound here before he goes to town. All right, so it goes a little film genesis. All right, it starts very fast, but these are actual sperm. Ejaculation has just occurred. All right, and the first thing that will happen is that you go this whole cloud of sperm going through the cervix into the uterus. This little part, I really don't know if it's animation or not, but it's so well done that it looks like it's real. And even if it is animation, it's very much like the real thing, right? Later on, it has to be photographs because the level of detail is just incredible. All right, so you see there's the cervix. Okay, now you see the sperm has just gone through the cervix. This is the inner lining of the uterus, the endometrium. At the very distal end there is the two openings of the fallopian tubes. These are the two fallopian tubes, so you're kind of going into a uterus up like this, all right? And this is all the mucus flowing down, the cervical mucus flowing down, covering the endometrium, and the sperm wiggling their way through the uterus into the fallopian tubes. The sperm is down here, it's kind of under the mucus, and it's gone into the right tube, all right? And continues. Oh, I want to point out also, so I mentioned functionally that it takes 100 million sperm to fertilize. You know, biologically, how many sperm does it take to fertilize one egg? Just one, okay? But functionally, it takes 100 million to consider a competent male. So it means that the selective pressure against fertilization is about 100 million to 1. That's the selective pressure against fertilization, which is just amazing. It's kind of similar throughout the mammalian world and in general, animals that reproduce sexually. And that's why, on average, it will be the fastest, strongest, most vigorous sperm that reaches the egg. It's called the most competent sperm, including the wiggling action of the tail and so forth, all right? So again, you can see that there's so much pressure on the sperm that, on average, it will be the strongest, most vigorous sperm that will fertilize that egg. And that's why most of us are kind of normal when we're born, because the abnormals were selected out, or just the less competent sperm never got to fertilize. That's one point. The other point is also that it's a very fast race. It can be anywhere from like 5 or 10 minutes after ejaculation to up to 5 days later, because the sperm in the female tract, for the human at least, lives up to 5 days. On itself, it only lives for about 24 hours, but the cervical mucus precisely carries, it's a very complex chemistry. It's a nutrient, it's a bactericide, it also is a lubricant, but it keeps the sperm alive up to 5 days after ejaculation. Another thing also is that half of the sperm, there's a chance process here. Half of the sperm never make it into the right tube, right? The other half goes into the wrong tube. And we know that happens with sperm because the sperm come from males and we never ask for direction. So half of that sperm is lost because they went into the wrong tube. Talk about stochastic. So that lost 50 million just went, it just eliminated 50 million from the competition. Yeah, never asked for directions. So this is going through the fallopian tube. At some point, they run into the villi, the hair-like structures of cells that are lined in the fallopian tube that are undulating. They're moving outward, right? So the sperm needs to keep being challenged and swimming against all that, and literally the current of the mucus that is flowing downward all the time. See the villi there? So fewer and fewer sperm are surviving this whole trajectory. You see at this point, it's only a few hundred or whatever. There's the egg, but it's not just the egg by itself. You notice that it's a whole cluster, it's a whole structure that has the egg inside, but then the two layers. The outer layer is the corona radiata, which is actually a cumulus of cells, and then the inner layer is the zona pellucida. All right, so here are some sperm that are arriving and are burrowing themselves through. There's a process called the acrosome reaction at the tip of the sperm. There is a capsule that contains digestive enzymes. The tip of the sperm is called the acrosome. So the acrosome reaction is when that tip makes contact with the egg, releases those digestive enzymes because the sperm literally has to digest its way through those layers in order to empty its nucleus and whatever little bit of cytoplasm it has into the egg itself, all right? So it'll show, there's going through the corona. Now it's going to show the acrosome reaction. I think it's on this one where it pours out the enzymes. There. And now it's going through the zona pellucida and it seals. The counterpart to the acrosome reaction from the sperm is a cortical reaction on the egg, that the egg seals itself literally within a fraction of a second. After the first sperm has gone through, the egg seals, and has to do with a calcium balance issue. And that way, no second sperm can come in, all right? Because within a fraction of a second, there's the second most vigorous sperm coming up. And if that sperm gets to empty its nucleus inside, what do we have? 23 from the egg, 23 from one sperm, 23 more chromosomes from another sperm, a chimera. It would be highly abnormal. That would code for 1.5 of everything, 1.5 brains, 1.5 3 arms, 3 legs, 1.5 of everything. So it's an extra load, an overdose of chromosomes, and that would be highly abnormal.
Is that XYY? Some males have XYY?
No, that's different. That's different. The sperm itself, the one sperm cell, the one sperm nucleus, has the extra Y.
Oh, I see.
All right? Or in the case of the egg, it could be that a female has an extra X. But that's in the gametogenesis process. All right, so now the egg has been fertilized and sealed, and it continues to travel down. It occurs in the fallopian tube, by the way. So here's now the sperm penetrating into the membrane of the egg, and it loses its tail, and then the membrane of the sperm digests, and the only thing that's left is now the nucleus, which is called a pronucleus because it's haploid. So a nucleus that is pronucleus, that is haploid, is called a pronucleus. This is the male pronucleus, and there's a female pronucleus inside the egg, and these two need to fuse. So there are at least 3 levels of fusing that you'll see or coming together. At the cellular level, egg and sperm, at the nuclear level, the male and the female pronuclei, and then at the chromosomal level also. So the male chromosomes need to line up with the female chromosomes to form the pairing, okay, chromosome pairs. So that's the female chromosome there. Now they're fusing together. Now we have already here, now we have a diploid number. We have a diploid cell. So this is no longer either egg or sperm, because by definition, egg and sperm are gametes, which are haploids. But now fertilization has occurred. And you see why it is so essential that the gametes be haploid. Because if the gametes were diploid and they fuse, then you're going to get a creature that is actually known as a tetraploid. It would have 4, and it would have double of everything. All right, you have a double load of chromosomes. Okay, so the fusion has occurred, the restoring of the diploid number. And now what it's going to show is the splitting of the replication, essentially. So this cell now is preparing to undergo mitosis, the first mitosis, within 24 hours. It will do a first mitosis. There. See? That's a chromatin, which are now going to, after replication, form discrete chromosomes. This, of course, is a diagram, all right? But it's showing the replication machinery, how it's going to split the DNA in half and duplicate it. And this is a diagram. All right, so that's the helicase, and then the polymerase that duplicates the chromosomes, and that's the chromosome pair. Each one of these is a chromatid, so these are sister chromatids held together by the centromere for each one of the chromosomes. Okay, and there are the other pairs. Now they're all going to line up singly. He's doing it very fast. They line up singly and then split into opposite poles. This is just straight up mitosis. Mitosis. This is straight up mitosis, the first mitosis, the first cleavage. And it will continue. I'm just going to let it run. You can see the embryo now. As the embryo is traveling down the fallopian tube, that embryo is already dividing. You can see the 4 steps. So first 2-cell stage, 4-cell stage, 8-cell stage. It's a duplicating number every time, right? Still within the fallopian tubes, because it will take 5 to 7 days for the little embryo to make the trip, to make the journey from fertilization in the ampulla, in the horn of the tube, all the way down to the endometrium. It will take 5 to 7 days, way after fertilization. All right, now the little embryo is going to kind of drop into the uterus, and you'll see the hatching process that I mentioned. In the meantime, the whole cluster is together, but the embryo continues to divide inside, see? Now it falls into the uterus, the endometrium, and there comes the hatching. Isn't that amazing? The embryo comes out of and leaves behind the corona radiata and the zona pellucida, because now they're no longer functional, they're no longer needed. Now there's a developing embryo before implantation, okay? That those 2 layers of cells have to be shed so that only the embryo implants. This is the endometrium here, the inner lining of the uterus. And this little embryo now has to do a little bit of magic, has to convince the endometrium that he or she is friend and not foe. Why? The endometrium is from the mother, right? And this little guy has 50% of his genes from the mother, but the other 50% come from the father. So this little creature, whatever it is ambiguous to the endometrium. The endometrium doesn't really know if this is friend or foe, if this is another one like it, or if this is a bacterium or some other organism that should not be in there, all right? And so the embryo has to convince the endometrium biochemically that he or she is a friend and not a foe, so that the endometrium allows this little creature to implant into the womb, into the uterus. And that all is done biochemically with very sophisticated signaling regulatory mechanism. Now it's just going to be showing the embryo itself without the surrounding for the sake of clarity. But the first stage is that the little embryo starts elongating at the top part of it, all right? Starts elongating, and then it will split in half, or it will form 2 halves, which is the bilateral symmetry. And then there will be a head end and a tail end, and that's the beginning of the formation of the embryo proper. The other part of the embryo is going to start embedding into the tissue of the uterus to form the trophoblast and the syncytial trophoblast, which eventually will become the placenta and vascularizing with mother. That's going down the neural groove, the center from head to tail on the embryo. That's the head region, and the back is the tail region. These are the somites. These are muscles that are forming along the spine. Already within about 3 weeks, the heart starts beating. The cells of the heart are self-beating. It's, we have 3 types of muscles. We have the skeletal muscle, the smooth muscle, which is unattached, and then we have cardiac muscle. The cardiac muscle cells have self-beating. They have a self-beating mechanism, and they're beating on their own like this. And when they start touching and joining to each other, they synchronize their beating. And it starts synchronizing so that when you have the whole mass of cardiac cells, like I said, within 3 weeks more or less of the fertilization, they're synchronized and they are beating together, and what will become the heartbeat of the embryo. Very fast heartbeat, by the way. I don't know if you've ever heard of ultrasound. I'm sure you've heard many of them of an embryo. Mhm. It's boom, 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 boom. It's like 180, 200 beats per minute. Very fast because there's, look at that. Yes, because there's tremendous activity going on here. Tremendous activity. Look at all the cellular division. The arms are pulling out. The digits have to be defined. Everything. The head, the brains. Look at the feet coming out. All the body parts are being formed very fast. Within the first 8 weeks, actually, the main body parts are formed, and the rest is just growth in size. Early on, we also have a little tail, which then gets absorbed, tail bud, and eventually gets absorbed back into the body. These may be ultrasounds that are colorized. Of course, in fast forward, okay? The baby's doing all kinds of things. He or she's, they suckle their thumb, they suckle their toe. They play with the umbilical cord back and forth. They can see some light going through the walls of the abdomen of the mother. All right, sometimes they open their eyes, but most of the time they're closed. Yeah, they can suck their toe. And it's starting to get crammed in there. They see light. They can hear sound also. Yeah, they play with the cord. What's this thing in front of me? So little Junior is very active in there and getting cramped because they're growing. I'll tell you in a minute what is the growth rate from the zygote to the baby at 9 months. In 9 months, that's dad there. That's mom just before giving birth. And here is the third. 2006, there she is. I think it's a girl, it's hard to tell. She gets her first bath. There, her first bath. That's the end of that. You know, so in 9 months, all this happens from a single fertilized egg, all right? Which by definition, a fertilized egg is no longer an egg because now that egg is diploid. So it's the first stage of the embryonic development. And that's where I wanted to land it. So let's look at that a little bit. I send you the link so you can see it again slowly if you wish, whatever. With the music, especially. Highly recommended. All right, a little bit of nomenclature again, because words are important and meaningful. It turns out that the fertilized egg is called a zygote, not only in humans, but in all animals that reproduce sexually, and even in plants. I would like to show the students because plants also reproduce sexually, right? The ones that do. What is the sexual organ of a plant? The flower. And the flower, so what is sperm in plants? The pollen, all that yellow stuff that we see covering everything in the springtime, that's pollen, that's sperm of the plant, you know, that's 100 million. And in fact, each pollen grain has typically 2 sperm nuclei inside, all right? And when plants fertilize, they also develop a zygote. And so it's universal. It's not just for the human or for mammals or for animals. Even plants, any plants, you see where it shows. A plant that is fertilized. Maybe on this one. As before fertilization. Yeah, but I'm just reading for the product. Oh, here, this is one. Okay. So just any standard textbook, if they're going to be honest, Here we show, close the image, that the fertilized egg is now a zygote. I had one. Here we go. Sorry. Oh yeah. This one here. How can I keep this one? All right, bigger. Okay. Here we go. Excuse me. Anyway, I don't know if you can see it there because it loses resolution, but it says there is zygote. Can you see it? Yeah. Okay, it says zygote. All right, this is a plant. So a fertilized egg, by definition, is a zygote universally in any species, including the human. All right, why am I emphasizing this point? Because zygote comes from the Greek, zygon, which means yoked or linked. And this is what I'm talking about, that the linking is at least at 3 levels. The linking or yoking is at the cellular level between egg and sperm, at the nuclear level between the male and the female pronucleus, and at the chromosomal level. In other words, the chromosomes from mom and the chromosomes from dad, all right? So that's the linking there.
Say that again, please.
The 3 levels of linking or yoking that occurs at the cellular level between the egg and sperm, at the nuclear level, in other words, the 2 nuclei, the male pronucleus and the female pronucleus, and then finally at the chromosomal level, the male and the female chromosomes, right? To restore diploidy. So 50% comes from the mother, 50% from the father. I'm repeating a lot. Here are some diagrams and photograph. So here's the egg with the zona pellucida, colonna radiata, all right? The nucleus, which is the haploid, this should be a pronucleus. These little granules are the calcium granules that are involved in the cortical reaction for sealing the egg after the fertilization occurs. Sperm, the structure is very different. The sperm is motile, so it has a long tail, a flagellum to move, and has a lot of mitochondria here, which are the energy factory for the motility of the sperm. Here's the acrosome that I talked about, and the nucleus, the haploid nucleus. This is an actual photograph, an electron micrograph, just to show the scale. This is 1/10 of a millimeter. So it's about 1/20th, the sperm is about 1/20th the size of an egg. Here's an egg with the zona and the corona, and here are sperm arriving. It's an actual photograph to have a scale view of the event. Moving further, this is the actual fertilization process in sequence. So here's the first sperm arriving, second one right behind it, gets sealed off. So the first thing, the acrosome reaction, digesting through the zona and the corona until the cytoplasm of the sperm is released inside the egg. And of course, the event of interest is to release the pronucleus, the male pronucleus, which will then fuse with the female pronucleus to form the diploid cell. So a zygote, by definition, is a diploid cell. It's the first diploid cell of us. We all started like a zygote. Here's a polar body, by the way, embedded in here between the zona and the corona, all right? And what else? Here's an actual photograph, another electron micrograph of the sperm surrounding an egg trying to fertilize. All these guys got left out because there's one that already fertilized and sealed the egg. This process of coming together of the 2 pronuclei, of the fusion of the 2 pronuclei, is called syngamy. And this is really what completes the fertilization process. You can see it's a process, right? It would take a few minutes to do this. And it ends with what is known as syngamy, which is restoring diploidy. This is when the male pronucleus fuses with the female pronucleus to make this a diploid cell. So we can talk about the process of fertilization, which chronologically may take a few minutes, but we can also talk about the moment of conception as a single event. You know, it's a little bit like eating. Well, I'm eating. When am I eating? When I start the soup or when I finish the dessert? Well, I'm eating through the whole process. Chronologically, there's a process, but there's also an event that is happening. And the event as a whole, fertilization or conception, are equivalent. You know, conception or fertilization. In the biological field, we say more fertilization, but people understand more conception. When are we conceived? Right?
That syngamy equals the conception moment.
Yes, yes. Well, the completion of conception. It's a process, but syngamy, once we have syngamy, then the two pronuclei have fused and we have a diploid nucleus. We have a diploid nucleus before first mitosis. Okay. So syngamy, we already have the diploid nucleus of the zygote. All right, let's back out for a moment and look at where fertilization occurs, because I'm just giving you detail here so that you can see it's a very biological, very definite process that is universal, that this is how it happens on every human that has been around, including ourselves. The normal place of fertilization is here in this area known as the ampulla. This is not to scale, obviously. If this were to scale, that egg would just be huge. But for a purpose of illustration, it's in this diagram. This part here is called the ampulla, or the horn of the fallopian tube. It's at the distal end of it. At the very, very end of the fallopian tube is known as the infundibulum, or the little hand, the little finger-like structures, the fimbriae. They come around the ovary. Notice that the fallopian tube is not directly attached to the ovary. There's a little sliver of space here, which I'll talk about. That's what causes the middle smirts, the middle pain during ovulation in some women, because that hand, this distal end of the fallopian tube, literally moves toward it's only about maybe a millimeter or two, but it moves toward the egg to capture the rupturing of the follicle. All right, so it's a movement there, and that little movement causes that pain, that little schmerz, which the Germans were the ones who discovered it. Anyway, here is the ovulation process. Here's the rupturing of the follicle, gets caught by the fallopian tube, and this is where fertilization occurs normally, all right? So that by the time the little embryo gets to the endometrium, 5 to 7 days on average have passed, and we already have a little embryo in process, even before implantation. Okay? And that's why, for example, a chemical that renders the endometrium inhospitable can be considered an abortifacient, because it is preventing the little embryo from being implanted. So it doesn't prevent fertilization necessarily, it is preventing implantation, which technically then is an abortion, okay? I'll get to that in a few more classes, but just to put it in context of the graph. All right, so the embryo itself goes through several stages, and these are the stages here. You can see here's the zygote. In fact, you can still see the male and the female pronuclei are distinct. They have not completed syngamy yet. These are microphotographs, actually. This is not a drawing or diagrams. These are micrographs. You can see this photograph is very illustrative. Here's the egg and the two pronuclei beginning to fuse, all right? You can actually see the polar bodies here also. Here's a polar body, here's another one. This is the zona, the corona, zona pellucida, corona radiata outside, okay? Then that cell, once syngamy is complete, that cell will undergo its first mitosis, cellular division. And this is the first cellular division here. This is a two-cell stage. It's still contained within the zona and the corona. You can see now the diploid nucleus in each one, distinct. Splits again into 4-cell stage. Splits again, 8-cell, and it continues to do that splitting. And until it reaches a stage, this is called compaction, because it maintains, because of the zona and the corona, the little embryo maintains his or her same size for the first couple of days. All right, the cells are getting smaller actually. So the cytoplasm, the overall cytoplasm is distributed into more cells. So each cell is getting smaller, and that is also called compaction, different from the DNA compaction into the nucleus. All right, it's also called compaction. All right, the next stage is the blastocyst. So what happens at the morula stage, at some point, this structure that has several dozen cells and still the corona and the zona as a cluster will hollow out on the inside. Will start hollowing out on the inside. Now, what's on the inside of that hollow? That's not just air, empty space in there, because there's no air to be had, okay? That's fluid. There's fluid inside, mostly water. But it does hollow inside like a little ball, like a tennis ball or something like that. That's hollow. And now we have the first layer of differentiation, of cellular differentiation, because the outer layer that is forming the ball, it's called a trophoblast, okay, has one function, and the inner mass that forms, the inner mass of cells, which is known just very unimaginatively as the inner cell mass. Sometimes you see embryoblast. The inner cell mass, or ICM, the ICM is a little mass on the inside of the ball. That little ball, that little mass, the ICM, is going to become the embryo proper. This little ICM will become the embryo proper. What about the trophoblast? What will the trophoblast become? Because the trophoblast is the one that is in immediate contact with the endometrium and with the inner lining of the uterus, then that becomes a placenta, the embryonic part of the placenta. The mother will also put its own side of the placenta with her own tissue, okay? But the bottom line is that the trophoblast will become the placenta, whereas the ICM will become the embryo proper. So we can say that when we get to the blastocyst, which is really the third stage, right? We got zygote, we got morula, and we got blastocyst. At this stage, there's already cellular differentiation, the first level of cellular differentiation. I want to go back to the morula for a moment, just for the sake of the word itself. I told you that zygote comes from the Greek zygon. Morula comes from the Latin mora, which, just like in Spanish, is a berry. Mora is a berry, like a mulberry, all right, or a raspberry that has little balls around it. But this morula is the diminutive, so tiny berry. Morula is the diminutive of mora in Latin. And so about a century ago, or a couple of centuries ago, when scientists were beginning to look at these structures under the microscope, of course, the scientific world at that time, and pretty much everybody in Europe spoke Latin, and say, oh, morule, morule. They were looking at little tiny berries under the microscope, and the name just stuck, but that's what it means, all right? It looks like a raspberry or something like that. Each individual cell is called a blastomere, and these cells are totipotent. They have the full potential to become a new human being. That's why identical twinning, when this guy splits into two, and they separate radically, they become two new individuals. Those are identical twins, monozygotic twins, all right? Because these cells are totipotent. By the time that we get to the blastocyst, they're no longer totipotent, they're pluripotent, because there's already cellular differentiation. You got the trophoblast, and you got the ICM, all right? So they've lost some potentiality. Still, if this blastocyst splits in half along the central disc, right, then not the central disc, the bilateral line, then you can still get two identical twins, even into the gastrulate stage, because the window for twinning, for identical twinning in the human, is up to 2 weeks, up to 14 days, which is already way into the gastrulate stage. All right, here is just a diagram of the embedding process that the dialogue has to go on, the biochemical dialogue between the embryo and the the endometrium to allow the embryo to implant and not be discarded. And the vascularization that occurs, of course, this area is highly vascularized. That's why when a woman has her period, what is happening is she's discarding an egg that was not fertilized, but she's also discarding the thickened tissue of the endometrium that became spongy, but there was no fertilization, no implantation, so that's discarded, and that's the bleeding there. Inside that blood and tissue, there would be one little tiny dot that would be an unfertilized egg. Here's the beginning of the establishment of the placenta through vascularization. And here is more organized and the little embryo continuing to grow. The fourth stage, generally globally, is called gastrula. And gastrulation, again, is a very important step because at gastrulation, then the ICM starts differentiating again and will form three basic layers or three germ layers. All right, the three germ layers of the gastrula. All of the organs and tissues of the body come from the three germ layers, all of them, all right? And so the three germ layers are simply how they are presented in the little embryo: endoderm, mesoderm, and ectoderm. And those are the three germ layers. For example, what's your intuition? The skin is going to come from the endoderm, mesoderm, or ectoderm. From the ectoderm, from the outer layer. Whereas bone and internal structure mostly are coming from the endoderm. And then muscle and other intermediate structures in the body are going to come from the mesoderm, right? Generally, except for the nervous system, which really, when you think about it anatomically, is the innermost part of the body because it's encased. Imagine to get to the brain or to the spinal cord, we have to go through skin, then we have to go through adipose tissue, then we have to go through muscle, then we have to go through bone. Finally, we get into the brain. It's the innermost tissue of the organ, of the body, and it comes from the ectoderm. Because of a very interesting process that happens during gastrulation, right, on the neural groove here, that's the word I was looking for. This is the head region, this is the tail region. These are actually micrographs, photomicrographs. What happens of the ectoderm starts pouring into the neural groove. It pours in, it's a process called invagination, okay? And the ectoderm starts pouring in like that, and eventually it will seal over and generate a tube inside, and that tube is going to be the neural tube, which will eventually become the nervous system, the spinal cord, and the head region, the brain. So it's a very interesting process how our nervous system really originates from the ectoderm, which is the same as the skin. Okay, here is again an intermediate, just a cross-section showing the syncytial trophoblast and how the embedding occurs, the vascularization and all that. Gastrulation, finally, I mentioned that within the first 8 weeks, these are the Carnegie stages, which are standardized now in Washington, D.C., and this is the standard models that are used for determining gestational age, how the embryo is developing and how the embryo is supposed to look like anatomically, externally, all right? And this is the first 8 weeks. You know, it starts with the follicle here and then fertilization, and it moves through gastrulation and early embryonic development. By the time we get to 8 weeks, which is 2 months more or less of the pregnancy after fertilization, not after first missed period, but after fertilization, all of the organs in the body, the major organs, are already formed, but in miniature. Okay, as you can see, even the external anatomy is fairly similar to the adult, generally to the adult stage. So all of the major organs internally are already in place, but they're in miniature because this little embryo is about maybe 1 to 2 inches long, all right, from head to rump. You can see the tail here. It's talking about the rump. See the tail very prominent, and then it gets reabsorbed. Vestigial, another evidence for common ancestors with animals that still have functional tails today. And from here forward, for the next 7 months, it's mostly growth in size. All right, more mitosis for all the organs to become bigger and bigger and bigger until he or she can be inside anymore. It has to come out at the 9th month. But from here forward, it's mostly growth in size. All of the major organs are, and tissues, of course, are in place. And so it changes name technically from embryo to fetus, which again in Latin, fetus means tiny being. And to illustrate that, this is just from, what's his name? It's a standard embryology textbook in medicine to this day. It escapes me the name. Anyway, this is a standard textbook that has been used for decades in medical schools for human embryology. The first 2 weeks, first 2 months is the embryo, and then from week 9 forward is changes to fetus, and this scale then goes into months here up until the 9th month, all right? And what I wanted to point out is that the biggest growth that we have ever in our lives is the embryonic growth. From a single microscopic cell, which is less than 1/10 of a millimeter in diameter, the fertilized egg or zygote, okay, 1/10 of a millimeter, to about a foot long when we're born, more or less, on average. Once I did the calculation, you know how many times we grow, how many factors? We grow about 5,000 times from a single fertilized egg, from the zygote to the time we're born, 9 months later. We grow about 5,000 times our original size. It's just amazing. For the rest of our lives, how much more do we grow? Maybe 5 or 6 times more, because let's say, again, for making it simple, the math simple, let's say that when we're born, we're a foot long. 6 feet is a tall person, right? So let's say 6 feet is the height, the highest that we're gonna get from the time we're born. So from 1 foot to 6 feet, we grow 6 times more for the rest of our life. But in the first 9 months, less than 1 year, we have grown 5,000 times our original size. It is just amazing how it works. And it's all automatic. We don't have to worry about it. We just have to come out and breathe for the first time. So the one major organ that is not functional yet until we are actually born is the lungs, right? The surfactant needs to develop enough to cover the alveoli so that it can take air instead of fluid. So this is the embryonic development. And I like to point it out precisely because of the ontological status of the human embryo. The ontological status, meaning at what stage, you know, are we more human or less human, or when do we go from non-human to human?
What time you were not you?
Right, each one of us, because we all started in this process. You know, we all started by a sperm of our father fertilizing an egg of our mother. And that's developing. So I hope that you can see that up until now, this is just biology. Whether I understand it or not, whether I like it or not, whether I agree with it or not, it's just the way it is. It's a biological fact, right? It's embryonic development. So to the question, when does life begin? At the individual level, ontogenetically, not the species phylogenetically, but ontogenetically, what's the most logical answer to when does life begin? Not only of the human, but of any species. You know, at fertilization. What else is there? In other words, before this stage, we got egg, we got sperm, which are cells from another individual. The sperm from my father, the egg from my mother, but in themselves, they're alive, but they don't have an entity as an individual. Without fertilizing, those two cells are dead end, they'll die. But at fertilization, there is this new entity, this new individual, all right, that in the species, in the human species, we call a new human. This is my argument anyway. If you agree or disagree, you can tell me and we can talk about it. But if we don't place the beginning of life here, when, at what stage you want to put it? When the tail is reabsorbed, when the baby is born, when you give him the car, when you give the teenager the keys to the car, when do we put the beginning of life?
The Romans used to put it like at 7 years or something.
Yeah, possibly. And the Greeks would put out their babies and across Mount Olympus to see who survived overnight and who didn't, right? Yeah, those in general are pre-scientific cultures. Today we have the biological evidence.
Well, if I'm not mistaken, the Jewish faith believes life begins at birth. So do they not have access to the same biological?
They do. So the question, exactly. So my question is why? What is it based on? See, because even to frame the question, when do I believe that life begins? It's not up to, what is my belief? My belief is in that God is a triune God, that God is three divine persons. I believe that we will go to either heaven or hell when we die, or maybe purgatory. Those are beliefs, but they're acts of faith. But even to ask, because remember, this is bioethics, so we have to do a little philosophy here. We have to do a little thinking. When I frame the question, when do you believe that life begins? That belief should not really be there. It's a misnomer, because it's not up to me where I believe it begins. It's a biological fact. I could say, let's say I say, well, do I believe that there are clouds out there? I don't believe there are clouds out there, but there happen to be clouds out there. So with all due respect, I just happen to be in the wrong because there are clouds. I don't believe there are clouds out there. Prove me that there are clouds out there. Well, look out the window. So I say, so it's a biological fact. Where does the belief fit in? If it were something as esoteric as clouds passing by, but here we're talking about life, human life. And the difference is between life and death, because it happens that abortion is legal in the United States up until the 9th month. So whereas these are anthropologically human beings, they have no protection under the law. And they can be killed legally with impunity. So, when we talk about human life, it's not just the color of the paint of the building that we're talking about.
Well, I think I disagree with your characterization that they're killed with impunity. I don't think that's a fair characterization. It may be a position that you believe if you want to have a debate, you can say it, but that's an extreme characterization. There are protections for the woman, there are protections for the baby or the fetus, and then there are protections and there's different values but to say with impunity, I think is a bit extreme.
Well, impunity in the sense that they have no legal rights, the unborn.
No, they do have legal rights.
What rights do they have?
Well, there's a compelling interest to say that a woman can't have an abortion after a certain age, after a certain point, unless there's other factors involved.
And what's the other fact?
It's not necessarily that a woman, without endangering her life or her mental health, the state can compel a woman to have a child.
Right, exactly. Actually, yeah, we're jumping into the next obvious topic, which is the topic of abortion, all right? I wanted to lay out the biological background first. But yes, we'll get into those arguments little by little. But the compelling, especially the mental, which is a loophole, right? Because the mental health of the woman. So could that be used as a loophole because I don't want this pregnancy? Because obviously it's an unwanted pregnancy. The wanted pregnancy will continue through, and sadly some people lose the wanted pregnancy, right? A miscarriage or a spontaneous abortion. But it's the unwanted pregnancy. How can we justify the unwanted pregnancy when we have a human being involved? Yes, I understand that the woman doesn't want it. It's extremely inconvenient. It will cause all kinds of distress in certain situations. But the reality of the argument for the vast majority is that it is inconvenient to have this child now because I didn't want to get pregnant. They weren't looking for the pregnancy, right? They weren't looking for it. I don't understand that part of it. And it's extremely inconvenient and distressing to have a pregnancy when I didn't want it. And it's also extremely unfair that it falls only on the woman because the guy walks away. Oh, that's not my child. That's all they have to say. It's not my child. And so they would walk away scot
Well,
clean.
with DNA testing, they can walk away so easily.
Exactly, but can you force the guy to have a DNA testing? You can't force it on him, right?
I don't know. You can't.
I don't think so. Well, if she's claiming paternity rights or something like that, she could force it.
Yeah.
And so there is some legal recourse to try to get the guy to have some level of responsibility on that pregnancy, right?
Well, if you walked away from me, I would want him to pay a penny, but that's just me. I
Right.
understand, and someone
I
will
know.
need the money, and I might need it too, but.
Yeah. The human complexity of the dynamics, we get it. We've been around enough to understand the stress of an unwanted pregnancy. Understood. But it's not really, at the end of the day, it's not life for life. And we'll look at life for life. Even when it's life for life, we can look at it.
Fathers walk away just because they choose, they have the option.
Pretty much
They walk
today,
away, the law doesn't let you walk away. There's a consequence. So the woman can't walk away. They can say,
She's locked
my
in.
body, my choice, I do it. I don't think that we have a choice. So there are subtle
there are,
subjects there, inequalities
Yeah.
in the law.
but even
And we
there,
see it from one angle. Definitely there's a lot to talk about it.
there is, and we'll get into this whole, one whole lecture devoted to abortion coming up. But also consider that even when the husband or the man is not allowed to walk away, that's one extreme, typically for paternity money, right, for money, but he cannot prevent the abortion either. When he wants the child, he cannot prevent the abortion. So where is the equal rights? It's his child too, but she's just stating it.
Before 1973, when abortion became legal, I don't know exactly what year ultrasound was developed. It was after that, but
Right.
I think that pre-1973 and for a few years after, there was some excuse for gee, I didn't really know how developed it was.
That's right.
But now,
Yeah.
I mean, when my older sister had, well, she's my cousin, but anyway, when she had her first child in 1980 something, 83, I think it was.
Mm.
It was very unusual to get ultrasound. I mean, she
Right.
had to like really ask for it because she wanted a picture and everything. It was a big deal.
Yes.
And now they're standard, at least
Right.
twice.
Yes.
So, even that's only like 10 years after.
Right.
I just don't know how you get around it. I don't know how you can look at that and deny that that's a baby.
Right.
Nobody, well, I wouldn't say they deny it's a baby.
Right.
That's not what people do, that's not what women do, that's not what
Well,
pro-choice
they
advocates do.
product of
Oh,
consumption.
Right,
sorry,
right,
you
right.
can, you can call it product of conception, you can call it a fetus, what was the other one? The fetus. I mean, you can call it what have you, but the fact of the matter is I don't know that a lot of people deny that it's not a human life. What they're saying is what is the moral balance and whose life, or you can say is more important, but what rights do you assign to one or the other?
Right. Right.
I mean,
Yeah.
that's the issue.
That's where we'll go. That's where
All
we'll go.
rights are not equal. There's a level of rights. Ones are more important than others. So it's not the same like the right of life than the right to speak freely. Those things are not at the same level. So one has to have
We're talking about a hierarchy of rights,
hierarchy of values.
of values.
Very few countries nowadays are prohibiting abortion, indiscriminate abortion, illegal abortion. So in Costa Rica, it's one of the few that remains. They establish like a reason, like a medical reason, biological reason, or I think that they even consider rape, pregnancy.
Rape or incest,
Right.
yes. Which is a very small
And
percentage.
you go through that process. It's not that complicated. You just need the medical opinion. Go to your gynecologist, and it is established that it's one or the other. And with the development in medicine nowadays, that's usually not a problem. So that is an excuse. I mean, there's a legal framework that allows you to do it at the beginning. But usually the things that are allowed in freedom is the abuse of it. It's like the responsibility part of the right that we usually don't get. And usually we debate about rights and rights and rights. And usually the people debating, they have a pretty good grasp of the issues. The problem is the outliers, the abusers of the right. I mean, what is at the other end? When you just open the floodgates and it becomes just a process. Like people abusing, like with the embryos and Planned Parenthood and the situations that have happened in some of those institutions about selling or making a profit out of stem cell research and all these things.
Well, the reality is that,
I see.
for example, the state of Florida, yeah, in the state of Florida that I'm more familiar with precisely through Respect Life Office, the vast majority, first of all, a woman doesn't have to give a reason for having an abortion. She doesn't have to give a reason. And the vast majority of abortions done in the state of Florida, there is no reason. She has given no reason. And certainly there's no medical reason and there's no life-threatening reason. It's the vast majority. That's the reality. In other words, the existential reality.
Well,
it truly is.
The vast majority. I mean, the reason is the vast majority because the vast majority of abortions are done in the first trimester.
In the first trimester.
Okay,
yes.
So then, later on, okay, so
right.
I mean, so I think, okay.
The people, the women tend to bond, right? They tend to bond with their baby from the second trimester on, so it's more difficult for the woman to actually have an abortion. She starts seeing the sonogram and all that. Anyway, this is kind of a setup. At least can we all agree that there's a biological process happening here that is standard, that is universal, that has nothing to do with my opinion in the sense that whether I believe it or not, this process will continue. It has been like this for millions of years and will continue foreseeably for the next millions of years if we continue as a species, right? There's a biological fact here. That's all I'm trying to establish in these first two lectures, that there is a biological fact, all right, that belongs to our species, and it parallels every other species, to the point that if there is an embryonic eagle, for example, inside an egg, I will be, if I disturb that egg, I will be put in jail, I can be fined and so forth. If it's an endangered species or a protected species, even at the embryonic stage, that embryo has protection under the law. More, way more than the human embryo. So there's a discrepancy in the social thinking here, that we will protect certain species even in their embryonic stage, and it doesn't translate over to the human. Those are facts that we're living with right now.
And if cellular life were found on Mars, that would be considered life. Microbes or whatever.
Yeah,
I mean, they would
if we wouldn't recognize
Mars,
it.
They would say that. So I don't see.
Right, I know. We have to struggle with this, and it's very important to maintain respect and civility in this conversation. Very, very important, because, I started in pro-life way before the priesthood. When I graduated from biology, I'll tell you the truth, when I graduated in biology from FIU in '73, and that's when Roe versus Wade happened. I was just in my early 20s. If you had told me I'm going to be a priest someday, I would have said I'll be a nun before I'll be a priest. I mean, it just wasn't in the horizon. My plan was scuba diving in the morning, sailing in the afternoon, and partying at night. Amen. That was the plan. Truly, honestly. And I saw it all of a sudden, Roe v. Wade. I said, what? It's now legal in a secular university. I studied at FIU. I studied at Miami-Dade 2.5 years, and then when I went to FIU 2 years. And I was studying biology. So everything that I had gotten scientifically, all of a sudden, it was legal. I said, how can this be? There must be a mistake. It must be that they don't know what they're talking about. And that's when I started getting involved in pro-life to try to explain to people, look, we actually have a developing human here. And since then, it's been 40 years, 50 years since then. I was still at it. I would hope that when we get into the lecture of abortion, we can discuss it in further detail and hear all of the possible arguments for or against and see, maybe, I don't know, maybe at the end of the lecture, we all become pro-choice. Okay, so what else do I have? Well, I have this other slide. Let me see, it's 12:30. All right, this is another one of these summary slides. Regarding embryonic development, there's a continuous development, right? So I'm emphasizing here just the continuity through the whole 9 months, so that at no particular stage we can say that we go from non-human to human or from less human to more human. If we did that, we'd have to say the same argument with a baby or a teenager or an adult and say that a baby is less human than a teenager and a teenager is less human than an adult. And yet it's not the case, right?
By the definition that you're using or the statements, adulthood defined biologically as a maturity of sexual organs?
Yes, the adult.
And capable of reproducing for a woman, ovulation, mature eggs, and capable of sustaining a pregnancy.
Right.
The definition?
This is biologically, adulthood.
So what is a woman after menopause? Is she no longer considered an adult?
Yes. This is the beginning of it, or I should put the beginning of adulthood.
Okay.
Yeah, just the beginning. In other words, an adult as opposed to a juvenile. We use those two words in biology. We talk about juveniles, the ones that are not capable of reproducing yet. The ones they're capable of reproducing, they have come into adulthood for any species, okay? And it's from there forward, of course. We may have, let me look at something here for a moment, because there's a little video that I could hold for the next class. It would help us frame. Next lecture, oh yeah. So we'll look at twinning and then abortion for next class. So yeah, let me hold that little video for next class. The next lecture is on twinnings, identical twins, and the whole ontological status. In other words, when does human life begin? When we talk about human, we have to include not just organically, but we have to include the soul also. All right, so the soul, the human soul. So we're going to talk about immediate ensoulment or delayed ensoulment in view of twinning. All right, because one of the characteristics of the human soul is that it is indivisible. The human soul is simple, indivisible, and eternal. We're going into the realm of belief here, of course, because this cannot be measured, all right? But it's the teaching of the church anyway, and few people have difficulty with that when they are believers. So again, just like the last course, the idea is to integrate our belief with the science and the facts of life. So if the soul is indivisible, how do we get from monozygotic twins, from identical twins, two individuals with two souls? They don't share half a soul each, right? And they're fully human. And think of just born twins walking around already. They have their own soul, of course, which is individual. So how do we get from one zygote, which received, let's say if it's immediate ensoulment, at the time of fertilization, they receive their soul from God. How do we get then two individuals with two souls? Okay, and did that one soul split into two or not? So we have to grapple with that, and that's going to be more at the philosophical and theological end of things.
You know, I talked to one woman, and she said something so bizarre. There's a lot of bizarre, but anyway, she said, a cloned human being would not possess a soul. I said, you know, thinking like that would make it very easy to just use clones for slaves or experimentation. And she was a professing Christian, and I was like,
Yeah, it's inconsistent. What the inconsistency is not at the biological level. The inconsistency is at the level of reasoning. All right, because essentially with a cloned human, if that clone survives, obviously the embryonic development and the implantation and all that, it also would involve in vitro because it's going to be done in the lab. Then that clone human would have a soul out of logical consistency. For example, if we believe in God and we believe that God is the author of life at the soul level, he's the one, so our parents can only create cells, right? Can only generate egg and sperm, but God generates the soul and grants us a soul when we're, let's say, when we're fertilized. All right, so one thing that God cannot be is cruel, because God cannot contradict himself, all right? So leaving a human being soulless, she's saying that this clone would be a human being that is soulless, literally soulless, then that denies that human being the possibility of redemption, of eternal life. Christ would be nothing to that human being. He would not be rescued by the blood of the Lamb. All right, so it leaves that human being out of the realm of possibility of eternal life and redemption, which is, I cannot think of something more cruel than God could do if we are God's image.
But isn't that the argument, and again, going back to New York Times, there's an op-ed about the concept of hell. I listened to Sister Jean saying that in her experience when she was down in Miami, she was talking that it does not seem possible that a loving God would allow someone to be tormented forever, and that our concept of hell, I don't know, I mean, this is my word because I can't remember what she, but it's sort of flawed or, you know, it's outmoded, but it does seem to be at least contradictory. That the concept of hell and a loving God are almost diametrically, logically impossible.
Correct. But yes, and so we say, at least in theology, we say it's not that God puts us in hell. First of all, notice the difference. I'm glad you bring it up because who's in heaven? People sometimes freak out when I ask who's in heaven, but it's a very simple, obvious answer, at least for believers, for us who are Christian and Catholics. It should be very automatic. Saints are in heaven. That's where they are. You know, where are the saints? Are they in hell? Are they in purgatory? They're in heaven. So all the saints, that the ones that we know, the ones that we don't know, and the ones that a church has canonized officially, and the ones that the church has not canonized, that's what it means. By definition, saint is a person who is in heaven for all eternity in the presence of the Trinity and other saints and angels, right? That's the belief. And we can name a few by name. How about John Paul II? How about Mother Teresa, St. Francis, your favorite saint? Okay, we could go back to Peter and the apostles, etc., etc. So we can name actually by name and time when they died and so forth, and when they lived, those who are in heaven. Quite a few. Who's in hell? Who would you like to put? I'd like to put a few people in hell, not really. In the moment of anger, I would like to put someone in hell. You know, typically Hitler comes up and a few others. There was a guy down in Africa, Idi Amin, who was just a carnivore, atrocious. You know, we can put a few, maybe some people will put the Castro brothers and the Southern Maduro and everybody else. But the church has never said a single name of a person in hell. The church has been very prudent. We pray for the souls in purgatory, okay? And I may have an idea of some people who are in purgatory, but the church has been very prudent never to name a single person in hell. Now, Dante took some liberties and put Muhammad in hell, one of the lower layers of hell. Dante Alighieri, the one who wrote the Divine Comedy in the Middle Ages, and that was a common belief. In the Middle Ages, the common belief was that...
quite a few people.
Yes, a few popes. He put bishops and popes in there because of shenanigans, all right? And he was... Yes.
Depending on where you were, and the flatterers and...
Yes, exactly. And he had all these layers, right, of the Divine Comedy. But that's just Dante's opinion, okay? That's not official church teaching. So the church has never really named anyone who's in hell. But it is still church teaching that there is a hell. Whether there's anyone in it or not, that's a different story. And I certainly don't want to be the first one. But you see, so there's prudence there. There's just prudence.
Mm-hmm. Mm-hmm. With the sages, with Aristotle, Plato.
Yes, right.
They consolidated, we put most of them.
Yeah.
In that, in the
Yes.
beginning. So we call
Yeah, but they didn't have baptism. You see, they didn't have baptism, so the pre-Christians had to go.
And I think the lowest level of hell is actually, aren't you upside down in the ice?
Is it? Oh my goodness.
Yeah, there's
So
one
none
that's
of
upside
that
down
is
in your
defined,
excrement.
though. I mean,
Yeah.
that is defined as separation from God. The fire and brimstone isn't defined, that's.
Right. That's colloquial. That's just to make it painful. That's to make it painful. That's the torment right there. That's the hell. But you see, and that's why how the church justifies it. It's not that God puts anyone there, is that these people want to be separated from God. The one thing that God never forces, again, God is very limited. He's a very limited creature, not creature, creator. God cannot force us into anything because he gave us free will. He gave us free will. The free will to reject him. And if we reject him eternally, if he could pain, he would pain for that. Because we reject him forever.
But it's also all merciful, and that's what you have.
Yes.
to hope for.
Exactly, and that's why I have a lot of hope in purgatory. My personal opinion is that purgatory is jam-packed full, and constantly people are going into heaven from purgatory. But there we
Isn't that why
finally
we say
learn.
prayers for them?
Sorry?
Isn't that why we say prayers for the souls in purgatory? That
Absolutely.
prayers.
Absolutely.
Oh,
Absolutely. And
You know, my
even
younger
though,
son got mad at me one time because we were having this conversation on hell, and I said to him, I know I'm weird, okay, but I don't even hope Hitler's in hell. I hope he
Of
repented and
course.
is living his eternity as
yes,
long as it takes in purgatory. He got furious.
right.
Never in my
Because
life have I ever
it's
told
a
person
it's
no.
a fracaso. How do you say
Because
fracaso? It's a
that's the worst thing
failure.
you can wish on them.
To have a single person in hell.
I know that.
Of
That's
course.
what you-
Of
Some
course.
people have this vision of Milton in
Paradise Lost.
Paradise Lost,
Better
that
to
you
reign
can
in
see
hell
how
than
the
serve in
angels,
heaven.
the angels that fell, they can tolerate the will of God and the order of things like that, and it's just out of that rejection, voluntary rejection.
It's the will. It comes down to the will.
Just don't want to do it, so they are banished themselves.
And that's why haughtiness or
That's what the devil says, Satan says.
Right.
Better to reign in hell than serve in heaven.
Right.
And that was the choice.
exactly. Yeah. See, and so it's ultimately it's a sin of arrogance, right? And it's a sin of pride and arrogance that is what can get us.
But even going back, saying it's a sin of pride or arrogance, in the face of total and utter evil, Hitler,
Right.
Pol Pot,
The Holocaust.
Stalin,
Mhm.
I mean, those are, on a grand,
Right.
on a magnificent.
Yeah, and the ones we don't see, the people who kill children and abuse children and so forth.
You've got to think the enormity of their evilness has got to be, at least seems to be manifest in the fact that they are mentally ill. That, so at some point, it's like Maggie says, she doesn't want, believe that even Hitler's in hell.
Right.
You have to,
No, you're on the right track. Shaban, listen to this. Look, in moral theology.
Evil is so premeditated and it's a process to life that you have so many opportunities to retool that thinking.
One would hope, yes, but people can get locked in,
You would
okay?
think like a Hitler,
Mhm.
like I said, now I think about it, well, obviously, like this is the magnitude of the evil would have to be mentally ill. But then you can take people who truly feel that way serious, maybe not to this, about like someone like a Henry Kissinger, who, had an objective choice to make and decided to, bomb innocent people in Cambodia. Well, that seems to be, I don't think he was mentally ill. I think he made a
Right.
calculation.
Right.
And so then you go, well, maybe that is pure evil. But then you could say, and again, the
Right.
arrogance, thinking he knew better.
Yeah. It does get into the topics of moral theology, which is the intention, the moral object, the circumstances, but also for sin, at least for grave sin or mortal sin, right? We talk about grave matter, full conscience, and full will. And
And if you
so
don't believe
it could
one
be.
of them, doesn't that, if you don't believe it, then it isn't a mortal sin? Isn't that
Correct.
what our church teaches us?
That's when we talk about objective and subjective. So we can talk about mortal sin objectively, let's say adultery, any one of the 10 commandments, right? adultery, murder, etc., mortal sin objectively. But subjectively, any one of us guilty or not, blameworthy, that's a different story, because now we're getting into the conscience of the person. That's why we don't judge the person. We can judge the act and say, well, objectively, this is adultery because that person is married to someone else. But subjectively, at the level of the conscience, we never go there. We don't have a right to go into a person's conscience. Only God can do that. That's the judgment. See? So subjectively, they may or may not be blameworthy. And if they are blameworthy, they may not be blameworthy at the level of mortal sin or grave sin. So guess what? They just avoided hell. Okay? So these are fine distinctions that have to be made. But at the end of the day, the conscience, we never go there. We don't have a right to judge the conscience. We don't know the person's conscience. Only God knows. And that's what we're
also
talking about.
taught where there is doubt, there is freedom.
Yes, right. But do we have a right? Then there is, we talk about speculative knowledge and evaluative knowledge. In other words, I may be in doubt about constructing a rocket because I'm not an astronaut or not an engineer, astronautical engineer, but I should not be in doubt with regards to biology or bioethics because I'm a bioethicist. I should not be in doubt with regards to the sacraments as a priest. I should know about the sacraments. If I don't know about sacraments, I can be held accountable. I cannot be held accountable for not knowing how to construct a rocket, but I can be held accountable for not knowing about the sacraments, the matter and form of every sacrament and so forth. So there is speculative knowledge. I'm not responsible for that. But there is evaluative knowledge. Evaluative knowledge is the knowledge that I need to make moral evaluation, to make moral assessment. And I can be held responsible for that, accountable. Exactly. And that's why the church, when the church always talks about conscience, it always talks about the formation of conscience, the education of conscience. And that's why, yes, there can be doubt, but we should not remain in doubt. That's the purpose of the whole course. We should not remain in doubt. If some women have a doubt whether their embryo is human or not, they should not remain in that doubt. They should clarify that doubt, especially if they're thinking of terminating that pregnancy. They should find out what that pregnancy is.
You keep saying that, like, I mean, I guess what I'm hearing is that women don't know that.
I'm giving them the benefit of the doubt that they don't know. I'm giving them the benefit of the doubt that they don't know. Why is it that why is it that the vast majority of women who hear the ultrasound walk away from the abortion?
I don't believe they do, but I don't know the statistics.
Well, but that's your belief, but I'm quoting a fact.
I'll try and see what I can find from Guttmacher or other statistics.
Yeah. Which, by the way, remember, Guttmacher works for Planned Parenthood, okay? So, caveat emptor, with a grain of salt. But it's true. They do. There's a Planned Parenthood outfit. But they are reporting what they want to report.
But if people who are in favor of abortion know it's a baby, then
How can you justify?
well, how do you justify? Yeah, how do you justify?
How do you?
Well, the same way that a desperate mother gets desperate with her newborn child crying and shakes the child until she kills the child because she's desperate.
I mean, that's wrong, of course, but that's
It's a mental frame. It is a mental frame. It's not only the mental frame.
I would suggest that under that reasoning, in the crudest sense, that basically makes the women a broodmare. And that their moral faculties are not really considered worthy because you basically say that this embryo is more worthy than the woman's life. And for you, Father, and you've said it a couple of times, and you describe it as an inconvenience. I think that term is loaded. Maybe in terms of civility, we need to find. I guess. I don't.
I'm happy to amend my words to the best way possible.
I think we have to give, and I don't believe the church has, and a lot of people give a lot of moral agency to the women, and so therefore, this is going. This is why this is probably one of the most fraught decisions, in our nation today.
Right.
I don't
Absolutely.
think that any woman jumps out of bed in the morning and says, "Yay, I'm having an abortion today." I don't believe that.
The typical scenario is that they're very distraught when they have it. In fact,
and I don't think anyone takes joy in it. I don't.
Right.
But at the same time, they're still doing something wrong.
You believe it's wrong. I believe other women don't believe it's wrong.
Well, you're saying, I believe, okay, but how can they not believe their child's life is not wrong? They do
what it really means.
She just said she
It has to
you
be
can
that,
totally
it has to
understand.
be that explanation, that they don't know what that really means. That they don't know that life has real values, that is an independent human being, that it could get to a full potential, and it begins from 0 to 60. And
Since
in between,
2020,
it's
I
the
can't
same
believe
thing,
people
just
don't
a
know
different
that's a
stage.
baby.
That process
Yeah,
doesn't
you-
exist, because what takes precedence is what part of my life is being affected. Without judging anything, I mean, it's not the embryo's fault. It's like, my life, I want to take it this way, and all of a sudden, this is an obstacle. And it's when you start seeing the embryo.
It's having more work than the woman's
versus,
life.
no, I would say baby versus obstacle. And you put those two elements and you compare what is the outcome, and you make a decision. My mom was 70 when she had me, and absolutely had the option. And it crossed her mind.
In communist Cuba, right? Which abortion is used as a contraceptive.
It was a mistake. My dad was 19. An absolute mistake. And embarrassing. He created a family issue, similar to Romeo and Juliet, something like that. And my mom, she wanted to be a doctor. I mean, she's a pediatrician. She ended up going to med school, but she had to take 1 year. She lost 1 year. It was all the social stigma of the early 70s.
Being pregnant out of wedlock.
She didn't. She decided, okay, not all of a sudden, I'm so happy that I'm going to have this baby. I'm so happy with this pregnancy. Absolutely not.
It was an inconvenience.
Absolutely,
You were an inconvenience.
I'm an inconvenience too. I'm sitting here. I shouldn't have happened.
It should happen, but it
I
happened.
shouldn't have continued more than a few days after she found out.
And this was-
And she can explain that. She said, that act, something told me that it's morally wrong. But there's no like a prefigurated knowledge, like she
Right.
was infused with the background to analyze all these things.
Right.
It was just completely wrong. What happens with all the other noise that you get, I'm taking the metro rail, and almost at every station, there's a sign that says, your body is yours. You can make the decision. And the photos are teenagers. And you can go and you call here. And one after one is that
It's a constant.
passive information. And all of a sudden you start making a judgment about that possibility. And it's just moving towards an angle and a decision. And only one sign about one woman, two life, only one I've seen in one of the stations. So there's not an equivalency. We're not having a dialogue here. There's an agenda. Somehow, that's the only way that you can explain the polarity of the message. Always towards get rid of it. Because it's always the easy thing. I mean, it's easy to go downhill, but going up. It's very easy to make a decision in an instant, get rid of it. Destroy is always easy. Build, like carry the child from that month, turn, and then you make a decision. Because unless it was in those exceptional moments of rape and health issues, 99% of the other are just a consensual, act. That doesn't happen by the Holy Spirit. That has happened only once. So you carry the turn, and then you give it away. Somehow, you don't want that baby, that's fine. That's not the issue, whether you have it or not. Whether you want to be a mom or not, is just complete the process because that's a human being. And then give it away, nothing. You're not going to be judged. You're not going to go to hell. It's a good thing. My mom could have done it. In the end, she decided to have me because my grandparents, 4 of them, said, big mistake, but the baby, we're going to help.
Stays.
We're going to help you. After all the issues that happened, there were moments.
They don't know what they were bargaining.
My grandfather, oh my God. You,
Now your grandparents are having second thoughts.
I mean, it's not easy,
Yeah.
but that's what is in my
It
mind.
is,
I'm
it
not
is.
judging anyone. I respect
It's a big, huge social issue.
completely,
Huge,
but
huge.
I have that opinion because of personal experience.
Mhm. Yeah,
I
and
think
this
I
was,
thank God I'm here.
this was in communist Cuba, right? Communist Cuba. And we know communist Cuba for decades now, because there's no money to buy anything, you know, women just can't afford contraception. And so abortion is used as a contraceptive method. It's called legrado. It's just a scraping. Go get your scraping and keep going. And if you get pregnant again, come back and we'll scrape it out again and keep going early on.
I mean, if that's not abusive of women, I don't know.
Absolutely. But the whole mentality is there. Cuba has one of the highest rates of abortion in the whole American continent, okay? Because it's used as a contraceptive method. And that's a reality. That's a social reality. It's a social reality. Yeah. So these are the issues that we have to deal with. Hopefully the background, the biological background helps some. Maybe yes, maybe not. I don't know. But from now on, we're going to get into the ethical issues, the bioethical issues, always backing it with the biology of it, all right? So at least we know we're speaking out of fact. We can still have an opinion, but at least we're speaking out of fact, out of knowledge. Folks, thank you very much. I know it's going to be interesting. We go there. Let's keep it respectful if we can, please. We must keep it respectful, but let's speak our mind, all right? Okay, so I'll save a little video for next time we come back, and we'll start with that. Thanks again, and I'm going to close this down, stop recording.